Okay, so welcome everyone to the 78th lecture of Dr. Hiley, Step 1. Before we begin, can we ask whether you guys can hear my voice? If you guys can I get a yes on the chat box, please? Okay, perfect. First and foremost, let me apologize for starting the lecture at 10 a.m. instead of 9 a.m. without any previous notice. It's because I was involved in a duty which said it would be finished by 8, but it took all the way till 9:15. So my apologies. It was extremely uncalled for, and we could not send you guys any email. So once again, I apologize. Hopefully, from tomorrow, duties of these sort will end at exactly 8:30, and I can continue the lecture at 9 a.m. Having said that, how are you guys doing? Did you guys complete your homework from yesterday? Okay, good. Okay, so from today, we are going to begin with pathology. And with pathology, the way that we want to move ahead is every day after we are done with pathology. Is it possible for you guys to watch an hour of Pathoma from Dr. Hussein and Sattar's videos? Everyone? Okay. So every day for the next 2-3 days that we read pathology, after we are done, please watch 1 hour of video of Pathoma. Okay. And that's that. And from the pathoma, I want you guys to focus on general pathology, the general pathology portion of pathoma. That's what I want you to focus on for the next 2 to 3 days. Okay, and you guys can work it out accordingly. Okay, so without any further ado, are we ready to begin the pathology lectures? Everyone, are we ready to begin the pathology lectures? Okay. Okay, so let's begin with the lecture. If you guys can see my screen, can I get a yes on the chat box? Can you guys see my screen? Okay, 1 second. Okay, perfect. So let's begin with pathology. So in pathology, the first thing that we have to understand is that with pathology, it's more about your understanding. That is, you have to understand what's going on in order for you to answer the questions perfectly. That's number one. Number two is from pathology, you have certain sections from where you will get tested, and we will tell you exactly which ones those are. So let's begin with cellular adaptations. I'm pretty sure we are all well aware of what cellular adaptations are. So first of all, let's talk about this over here in a short discussion. That is, cellular adaptations are basically, an adaptation is basically a change in the cell, in the physiological or in the structural portion, after which if the cell is exposed to an unfavorable environment. Okay, if the cell is exposed to an unfavorable environment, the cell can undergo some physiological and some structural changes which would allow the cell to survive. So cellular adaptation is basically the adaptation of a cell in order for the cell to survive. For example, if we talk about cellular adaptation in terms of hypertrophy and hyperplasia, hypertrophy, let's say that muscular hypertrophy. Let's say when you go to the gym and you work out and you put your muscle through an excessive load, right? When you put your muscle through a lot of load, if your muscles do not increase in size, not in number, if your muscles do not increase in size, that is hypertrophy, there is a possibility that the next time when you increase the load on that muscle, is there a possibility that the muscle can tear away? Yes or no? Right? So that is hypertrophy. So the thing is, when your muscles realize that you are putting your muscles through so much stress, and if the next time you go do the same thing, and if it doesn't increase in size, then there's a possibility that when the muscle undergoes that kind of stress once again, the muscle can tear away. So that is hypertrophy. Hyperplasia is increase in number of the cell. Hypertrophy is increase in size of the cell. Hyperplasia is the increase in number of the cell. Hyperplasia basically is when the cellular number increases in response to a damage. For example, hyperplasia is, let's take the example of endometrial hyperplasia. For example, endometrial hyperplasia is when the uterus is exposed to an extreme amount of estrogen. For the first stage, it could undergo a little bit of hypertrophy here and there, but when it realizes that with the increased amount of estrogen, okay, it's stimulating the number of the cells. What happens is instead of increasing in size, it increases in number, okay, so that it can prevent any other further damage. So hyperplasia is also another type of, it's a pathological hyperplasia. Does anyone have any idea of a physiological hyperplasia? Physiological hyperplasia? Does anyone have any idea of a physiological hyperplasia? Pregnancy, very good. Okay, so for example, pregnancy is a physiological hyperplasia. So can we assume that hyperplasia is a controlled proliferation and differentiation of the cell instead of uncontrolled proliferation and uncontrolled differentiation, which is known as carcinomas? Can we assume that hyperplasias are controlled proliferations of a cell? Yes or no? Can you guys hear my voice? Controlled proliferation of the cell, hyperplasia. Okay, I would really expect some faster feedbacks, please. Okay, if you want to finish this today. So if you guys have any questions, please let me know. Please ask me the questions after we are done with a single page. I'll ask you if you guys have any questions or not. I'll be more than happy to make you understand this. So we're done. So hyperplasia and hypertrophy. The next one, the next kind of cellular adaptation that we want to talk about is atrophy. Atrophy is basically the decrease in size of the tissue mass. This could happen because of lack of blood supply. This could happen because of lack of working. This could happen because of lack of your nutrients or oxygen, for example. That's that. Metaplasia, we all know that. So we are done with hyperplasia, hypertrophy, atrophy, and now we're going to talk about metaplasia. Metaplasia is basically the change of one type of cell to another type of cell. Please give me a very fast example of metaplasia. What is the most common example of metaplasia? Barrett's esophagus, very good. Barrett's esophagus. What happens is that in Barrett's esophagus, we have the transformation. Okay, we have the transformation of the which is the malignant transformation, where the Barrett's esophagus can actually give rise to esophageal carcinoma, where what happens is you have one type of cell and that changes to another type of cell. So basically, you have the epithelial cell that is there in the esophagus. Is it a simple layer or is it a stratified layer? Fast answers, please. Is it a simple layer or is it a stratified layer? Okay, so it's a stratified layer. Basically, by the word stratified, we mean that one layer is on top of another and then on top of another. So when you have any insult to the esophagus, for example, long-term cigarette smoking or alcohol, the layers, they keep on depleting, and then at one point the epithelial layer, it realizes that it needs to change its structure in order to survive. It needs to change its structure in order to survive. So what it does is there is a change of the normal epithelium to a columnar type of epithelium. This is known as columnar metaplasia. And this metaplasia, isn't there a possibility that this, if the cells are big, if the cells have become metaplastic, if you do not stop the insult, there's a possibility that this metaplasia can actually full on change to dysplasia? Yes or no? Yes. So dysplasia is, so what is dysplasia? Dysplasia is not anaplasia. Dysplasia is the loss of normal cellular architecture. What happens is, if the cells, they become dysplastic, for example, are you guys well aware of leukoplegia? I'm pretty sure you guys know what leukoplegia is. Do you guys know what leukoplegia is? Leukoplegia, right? Every one of us should know what leukoplegia is. Leukoplegia, erythroplegia, these are precancerous dysplastic lesions that happens in the buccal mucosa in long-term cigarette smokers. So leukoplegia, these are actually not anaplastic cells, these are dysplastic cells, meaning that they have lost their architecture due to long-term insult. So previously we could have assumed that there was hyperplasia, then there was metaplasia, then there was dysplasia, and now the patient has full-on leukoplegia, which is a dysplastic feature. And what happens is, if the patient has dysplastic changes, if you do not stop the insult now, then there is a possibility that this dysplastic cells can change into anaplastic cells. And anaplasia is what we know as, is what would change into neoplasia. Neoplasia is carcinoma. So cellular adaptation, once again, cellular adaptations, these are reversible changes that can be physiological, for example, uterine enlargement, or pathological, for example, myocardial hypertrophy due to systemic hypertension. Why do we have myocardial hypertrophy and systemic hypertension? Why do we have myocardial hypertrophy and systemic hypertension? Increased load. Increased which load? Preload or afterload? Afterload. Very good. So afterload is increased. As a result, the left ventricle has to work excessively to push the blood out. So the left ventricle undergoes hypertrophy. What kind of what kind of a heart failure would this lead to? Systolic heart failure or diastolic heart failure? Very good. This would lead to a systolic heart failure. Can anyone tell me an example of a diastolic heart failure? Diastolic heart failure. Diastole, where the heart cannot relax. I mean, systolic heart failure. My apologies. My apologies. Systolic heart failure, not a diastolic heart failure. DCM, very good. Then what else? Okay, so basically, so these are the changes that happens due to, for example, you have the physiological one, that's that, and then another one is the pathological one, which is the myocardial hypertrophy due to systemic hypertension. If stress is excessive or persistent, adaptation can progress to cell injury. Okay, so we have a question. Can you rearrange left ventricular heart failure, diastolic or systolic? Of course I can. Just let me finish the page and then I'll answer the questions, okay? Just give me 1 second. If stress is excessive or persistent, adaptation can progress to cellular injury. For example, significant left ventricular hypertrophy can result to injury to the myofibrils and heart failure. Now, let's talk about hypertrophy. Increase in structural proteins and organelles result in increased size of the cells. Okay, so what you have to understand is that hypertrophy is increasing size of the cells, and this example is cardiac hypertrophy. Over here, this is tested that whether you guys are well aware of the terms that is hypertrophy is for this one over here. Okay, so that the increase in structural proteins and organelles and increase in size of the cells, that this is known as hypertrophy. This is a question. Okay, so are the same for all of these ones. Basically, the questions are going to come from over here. The answers are that whether you know the terms or not, that is the answer. Hyperplasia is controlled proliferation of stem cells and differentiated cells, and increase in the number of cells. So that is hyperplasia, but you have to focus on this word over here, that this is a controlled proliferation. The proliferation is controlled over here. Excessive stimulation can result in pathological hyperplasia. For example, we said the example of estrogen and uterus, where excessive amount of estrogen can result in endometrial hyperplasia, and this may progress to dysplasia and carcinoma because it's excessive stimulation. So that's that. And another example is BPH, benign prostatic hyperplasia, under the stimulation of dihydroxytestosterone. So that's that. Atrophy is the decrease in tissue mass due to decrease in size. This is high yield. I'm pretty sure we discussed this in the UWorld notes previously, that in atrophy, you have cytoskeletal degradation by the ubiquitin proteasome pathway and autophagy. That is, there is decrease in protein synthesis. So cytoskeleton degradation via ubiquitin proteasome pathway. What happens, I'm going to talk about this once again. That is, do you guys remember we talked about this, that in, you guys remember that in a carcinoma, we said that there is muscular cachexia by the process of ubiquitin proteasome pathway under the influence of tumor necrosis factor. Yes or no? Who remembers that? Right? What happens is there is muscle breakdown because the muscles get tagged by this protein over here, that is ubiquitin. Once you tag a protein or you tag a structure with ubiquitin, this has the tendency to go to the proteasome. Proteasomes are basically the organelles of the cell that are responsible for containing a considerable amount of proteases that can break down large protein structures. So basically, if there is a tissue mass that has to decrease, there is cytoskeletal degradation via tagging by ubiquitin and then going to the proteasome pathway. This is tested multiple times in NBME, ubiquitin proteasome pathway, multiple times. This is tested in NBME. So do not forget that if anything has to decrease in size in your body, the process by which it's happening is ubiquitin proteasome pathway. And also, there's also apoptosis. The apoptosis is decreasing the number of cells. So that's that. It includes disuse, disuse atrophy, denervation atrophy. Disuse atrophy, meaning that, for example, if you see a patient who has recently broken a leg and his legs are in a cast for the last 3 months, you'll realize that the muscle on the, for example, if it's a left-sided cast, his left-sided leg muscles are much smaller than his right-sided leg muscles. Basically, if you do not use a muscle, they will undergo atrophy. So disuse atrophy, that's that. Another one is denervation. That is, if the muscles lose nerve supply, right? If the muscles lose nerve supply, lack of stimulation to the muscles results in muscular atrophy. That's that. Also, loss of blood supply is obviously there. Then you have hormones and poor nutrition. So loss of hormonal stimulation and poor nutrition. That's that. Okay. Next one. Next one is metaplasia. Metaplasia is reprogramming of the stem cell. Is it not necrosis when lack of blood supply? Lack of blood supply, when there's absolute lack of blood supply, the cells can undergo necrosis. When there is considerable decrease in blood supply, then the cells undergo atrophy. Are we clear? Okay. Metaplasia. Metaplasia is basically the reprogramming of stem cells, basically where one cell type is replaced by another type of cell to adapt to a new stress. This example goes once again. For example, in the respiratory tract, when we are smoking for a long period of time, the cigarettes, they go and they attack the cilias. The cilias are basically the microfilaments that are responsible for beating left and right in order to push the mucus from the respiratory tract all the way up to your esophagus, so that it can get digested away, right? Or you can spit it out, or you can cough it out, or you can sneeze it out. So whichever. But the thing is, if there is a person who has a long smoking history, that cigarette smoke will destroy the ciliated epithelium, right? And with long-term damage, what would happen is you would realize that the respiratory ciliated columnar epithelium is changed by squamous epithelium. So there's squamous metaplasia. Basically, the reason is when they're exposed to this kind of injury, the respiratory tract, they change from ciliated to squamous in order to prevent further damage. As a result, what happens is that's why when there's squamous epithelium, the mucus is not beaded all the way upwards by the cilia because there's lack of cilia. So the mucus accumulates for a long period of time, and that's the reason why cigarette smokers are more prone to develop pneumonia, bronchiectasis, and all the other types of diseases. So that is metaplasia. So it's usually due to exposure to an irritant. For example, in Barrett's esophagus, there is in Barrett's esophagus, when there is excessive amount of gastroesophageal reflux disease, this can give rise to the change that is from squamous. You have the columnar metaplasia, right? And then in the respiratory ciliated epithelium in exposed to cigarettes can change from columnar to stratified squamous epithelium. So that's that. And there is a huge possibility that if there's a dysplastic feature, if the injury is not stopped then and then malignant transformation will happen, considering there has been persistent insult or injury. That's that. For example, once again, Barrett's esophagus, if it's not stopped, if you do not prescribe PPI right away, and the patient does not undergo diet and lifestyle changes, then there's a huge possibility Barrett's esophagus will turn into esophageal carcinoma. So that is that. Okay. Dysplasia. Um, Dysplasia is okay. This is what I want you guys to focus on for 1 second before I go to dysplasia. Myositis ossificans. This is a comparatively newer focus of USMLE Step 1. Okay. This is a comparatively newer focus of USMLE Step 1. I don't know why they are a little bit obsessed with myositis ossificans. Basically, what happens is the muscles are converted to bones. So what happens is over here, the patients, they um, they complain of excessive myalgia. Okay, they complain of excessive weakness, tiredness, fever, fatigue, myalgia. And what happens is that when you do an X-ray or when you do a CT scan, but on X-rays, what you see is that on the muscles, you will see small depositions of this white crystal-like calciums. So if you see any sort of X-ray presentations like that, please try to remember that myositis ossificans is the conversions of the muscle cells into bones or into calcium-like structures. Are we clear about this? Yes or no? Are we clear about this? Okay. Especially there could be a history of trauma. Okay, there could be a history of trauma. So what would happen is, for example, the patient can complain that recently the patient was involved into some sort of an accident, number one, or the patient that was involved in some sort of fight or something like that. Okay, or something where there was trauma to the muscle, and after trauma to the muscles, the muscle cells regenerated with depositions of calcium in it. So regenerations of muscles after trauma with depositions of calcium is known as myositis ossificans. Okay, so that's that. Please don't forget this. This is a newer obsession, and comparatively, you will find a lot more new questions with myositis ossificans. This process is this process is a metaplastic process. This is a metaplastic process. Okay, dysplasia. Let's go to dysplasia. Dysplasia is disordered precancerous epithelial cell growth, not considered a true adaptive response. Basically, you have basically the only thing you should remember is the cells have so much to tolerate, right? The cells will undergo hypertrophy, hyperplasia, they're still tolerating, then atrophy is there. Then there is metaplasia where they get angry a little bit. And dysplasia is when they get really angry. Do you understand what I mean? For example, when you expose the cells to a long term of injury or insult, then they get really angry, and then you have disordered precancerous epithelial growth. Okay, precancerous epithelial growth, not considered a true adaptive response. Basically, this is not a true adaptive response. This is characterized by loss of uniformity in cellular size and shape. Basically, this is called pleomorphism, meaning variation of shape and size, and loss of tissue orientation or tissue architecture. Now, nuclear changes over here, what happens is in the nuclear changes, you have increased nuclear to cytoplasmic ratio and clumping of the chromatin. What does this mean? This means that in a normal cell, the nucleus Okay, the nucleus should be smaller than the cytoplasm. If the nucleus is smaller than the cytoplasm, then the nuclear to cytoplasmic ratio, should be low or normal. Okay, meaning that the cytoplasm should be bigger than the nucleus. If the nucleus starts enlarging in size, okay, if the nucleus starts getting bigger in size, this shows that there is a lot of proliferations of genetic materials that's going on over here, and this can actually stimulate the cells to grow in an unproliferated manner. So increased nuclear to cytoplasmic ratio is a sign of dysplasia, and also, to be honest, it's also a sign of an anaplasia. Painful, firm, mobile mass within the muscles. Yes, that's myostatus ossificans. Okay, painful, firm, mobile mass within the muscles. But basically, for step one, you will have questions not regarding painful, firm, mobile masses. You can have questions regarding X-ray findings. Okay, so in X-ray findings, if you see calcium depositions in the muscle, a lot of students will write the dystrophic calcifications. Okay, but dystrophic calcifications are absolutely the wrong answer because they happen in dead necrosed tissue. If there's calcium deposition in previously healthy traumatic tissue, then this is myostatus ossificans. Okay, now what was I saying? I was talking about nuclear to cytoplasmic ratio and how they are a sign of anaplasia. Does anyone know the other signs of anaplasia? Increased nuclear to cytoplasmic ratio is one sign of anaplasia. What are the other signs of anaplasia? Fasten. Guys, can you hear my voice? What are the other signs of anaplasia? Increased nuclear to cytoplasmic ratio is one sign. What are the other signs? Can you guys hear my voice? Okay. Are you guys angry that I started the lecture at 10 a.m. and now you're not talking? Is that what it is? Okay. Okay, thank God. Okay, all right. Okay. So are you not talking because you don't know the answer? Okay, no problem. Okay, good. No problem. Or then just remember that increased nuclear to cytoplasmic ratio is one feature of anaplasia. Okay, basically, if you're a pathologist who is viewing a cell under a microscope, how will you know if the cell is a normal cell, or how would you say that this cell is a anaplastic cell? First of all, you would see the nuclear size. You would see the nucleus size. You would see the size of the cytoplasm. If the nucleus is um, bigger than the cytoplasm, then you have increased nuclear to cytoplasmic ratio, and this is a very high yield sign for anaplasia. That's number one. Then number two is clumping of the chromatins. If the chromatins get clumped, that's another one. Okay, then that's another one. And another one is if you have increased if you have in is that the same as mitotic ration? No, I'm not sure what mitotic ration is. Okay, but anaplasia is basically if you're a pathologist, if you have to see the cell under the microscope, then these are the things that you should look out for: increased nuclear to cytoplasmic ratio, clumping of the chromatins, then depositions of nuclear inclusions, right? Cells shape and size of the cell, nuclear pleomorphy, and cellular pleomorphism. So they have mentioned some over here. They have mentioned that there is cellular pleomorphism. They have mentioned increased nuclear to cytoplasmic ratio, and they have mentioned clumping of chromatin. The reason why I'm discussing these properties right now is because I'm just letting you know that by the time the cells are dysplastic, they already have the features of anaplasia. That is, they already have features of anaplasia with cellular pleomorphism, increased nuclear to cytoplasmic ratio, and clumping chromatin. These are the 1, 2, 3 features of anaplasia already. Then mild and moderate dysplasia, which will not involve the entire thickness, may regress with elevation, meaning that if you move away the inciting injury or inciting insult, the cells may regress to an initial normal cell. But if there's a severe dysplasia, this may progress to carcinoma in situ. Fast answers: carcinoma in situ. What does that mean? What do we mean about carcinoma in situ? What do we mean about carcinoma in situ? Very good. Without penetrations of the basement membrane, that is carcinoma in situ or T0. It is usually preceded by persistent metaplasia or pathologic hyperplasia. Okay, I feel like we have talked enough about cellular adaptations. Okay, fast answers. If you have a cell that has increased in size, what is the term for this? Fast answers, please. Cells that have increased in size, what is the term? Hypertrophy. Cells that have increased in number, what is the size? Hyperplasia. Okay, cells that have decreased in size due to denervation, disuse, decreased oxygen supply, decreased nutrition, decreased blood. Okay, next one is, if there is changes of one type of cell to another type of cell, what is the name of the term? Metaplasia. Then the next one is, if you have disorganized or loss of cellular architecture, what is the term? Okay, now, so we're done with this page. We're going to go to the next page. What questions do you guys have from this page? Dr. Hussam, I remember you had one question. What is your question? Who else has questions? Does anyone have any questions, or should I move forward? Okay, is aplasia and apoptosis the same thing? First of all, aplasia is, we did not discuss anything called aplasia as of yet. Aplasia is absence of a cell, okay? Do you mean anaplasia or do you mean atrophy, Dr. Medical? Anaplasia. Anaplasia and apoptosis the same thing? Absolutely not. Anaplasia is predisposed. Anaplasia is basically carcinoma. Anaplasia means that the cells have that there has been previously the cells had dysplasia. Now the cells have anaplasia, meaning that the cells have uncontrolled growth and uncontrolled proliferation. This is the pathological term for carcinoma, anaplastic. Okay, that is anaplasia, loss of cellular differentiations. If the cells are well differentiated, okay, we'll talk about this in the future. Cellular differentiation, basically, if the cells are well differentiated, that means that the cells are very similar to the normal cells. For example, squamous cell carcinoma is a well differentiated carcinoma. If the cells are not differentiated or not well differentiated, this means that the cells are very different from the normal cell of your body. For example, you have adenocarcinomas. These are not very well differentiated cells. So that depends on the grading of the carcinoma. That is anaplasia. Atrophy is, wasn't that your question? Sorry, anaplasia and apoptosis. Apoptosis is basically cellular death. Apoptosis is basically cellular death, and the process of apoptosis is something that we will talk about in the future in 1 second. That is, you have cellular blebbing, and then the cell kind of dies because of release of intracellular enzymes and caspases. So that is apoptosis. Okay, so that answer was for Dr. Medical. Next one was for Dr. Hussam. NC for dysplasia and carcinoma. What do you mean by NC? Okay, while you explain to me what NC is, I'm going to go to Dr. Karbasi's question. My side is ossification. Can you explain one more time? Yes. When a muscle undergoes trauma, there's a possibility that the muscles can get regenerated with excess deposition of calcium in the muscles. Basically, we know that calciums love to deposit themselves in alkaline mediums. So if there is, for some reason, there is There is muscular regeneration. This doesn't happen to everyone. Not everyone gets their muscle regenerated with deposition of calcium. Some people, they have excessive predisposition of an alkaline medium in the muscle regeneration process, and this invites a lot of calcium to come and deposit when the muscles get regenerated. So you have this muscle regeneration with depositions of calcium. So when you do an X-ray, you find small dots of uh, calcium crystals in the muscular region. This is known as myositis ossificans. Myositis for muscle inflammation, and ossificans for deposition of calcium. That was for Dr. Karbasi. Next one is Dr. Osam. Okay, nuclear to cytoplasmic ratio. Nuclear to cytoplasmic ratio for both dysplasia and anaplasia. Basically, nuclear to cytoplasmic ratio is the increase in size of the nucleus compared to the size of cytoplasm. Basically, the nucleus to cytoplasmic ratio should be at least 1:1, or the cytoplasmic size should be more than the nucleus size. If there's a cell with an increased size of the nucleus, we can assume that there has been uncontrolled proliferation and uncontrolled growth of the genetic materials. This can give rise to uncontrolled protein-like structures in the future. So increased nucleus to cytoplasmic ratio is a very high yield sign for dysplasia and anaplasia. Basically, if you are a pathologist in the lab trying to understand whether the cell you are viewing under the microscope, is it a normal cell or is it a anaplastic cell? The first thing you will see after seeing the cellular size and shape, that is pleomorphism. If it's there, then you would see the nuclear size, and then you would compare it with the cytoplasmic size. If the nuclear to cytoplasmic size ratio is high, then this is a very important indication that the cells could be anaplastic or carcinogenic. Am I clear? Have I made myself clear? Everyone? Does anyone have any question, or can I move on to cellular injury? Okay, let's move on to cellular injury. I'm going to talk about this. If anyone has any question, please keep it to yourselves. And after the end of this page, I'll ask you guys whether you guys have questions, and I'll answer it accordingly. Cellular injury. First of all, we had cellular adaptations. Basically, what happens is, let's say someone comes and keeps on annoying another person, and that person keeps on changing, keeps on changing. And then, for example, all of a sudden, that person does not have the ability to change anymore, so the person just dies. So that is cellular injury. Basically, what happens is you have an injury or an insult. You keep the injury or insult keeps on attacking the cell again and again. So the cell undergoes hypertrophy, hyperplasia, metaplasia, dysplasia. And then if the cells do not undergo anaplasia, and for example, at the level of dysplasia, this is what you can understand. At the level of dysplasia, two things can happen. Okay, if you do not remove the injury. Okay, so the cells, in order to be dysplastic, they have previously been hyperplastic, they have been metaplastic, now they are dysplastic. If you do not remove the injury, or if you do not remove the insult, there are two things that can happen right now. First of all, the cells could be anaplastic, that is, the cells can get very angry and have unproliferated carcinoma-like growth. Another one is if the cells can die. Okay, cells can die. This is basically cellular injury. Now, this cellular injury over here, this could be of two types. That is, this could be reversible. That is, the cells, they will die, but they can regenerate again. And another one is irreversible. Irreversible is basically the cells have died in such a manner that cellular regeneration is impossible. So let's talk about the first one, that is reversible injury. What is reversible injury? Reversible injury is basically the loss of function, but then loss of function of a cell, but the cells gain the function back. Reversible injury is characterized by, first of all, it's characterized by cellular swelling. Always remember this, cellular swelling. And along with cellular swelling, you have swelling of mitochondria. Okay, whenever you hear about swelling and injury, think about reversible cell injury, that the cells have undergone reversible cell injury. Basically, the reason why cellular injury, cellular swelling and mitochondrial swelling happens is because the inciting incident attacks the sodium potassium ATPase pump. sodium-potassium pump, which is responsible for maintaining the osmotic balance inside and outside the cell. If there is loss of this kind of a pump, or obviously there's an osmotic imbalance. As a result, there is cellular and mitochondrial swelling. And when cellular and mitochondrial swelling happens, especially mitochondrial swelling, you have what we say is the ATP of the cell decreases. So you have decreased ATP. Okay. Now, so cellular and mitochondrial swelling, that's one. Okay. Another one is you have changes in the plasma membrane. You have changes in the plasma membrane. So what you have is you have these cells which undergo small protrusion. So for example, if this is the cell, and now with injury, there has been laxity of the plasma membrane. So small portions of the cytoplasm actually blebs out like this, okay? It actually blebs out like this. This is known as cellular blebbing, okay? That's that. And another one is there is small amount of clumping of the nucleus. So there is chromatin clumping, okay? Clumping of the chromatin. Chromatin clumping, basically the chromatins, they should be very nicely packed in the nucleus away from one another at a normal physiological distance. But when there is cellular injury, what happens is they get scared. Okay, for example, if this is John and this is Jane, then they get scared and then now they want to be together. So what they do is they just clump together. Okay, so just don't forget about your friends, John and Jane. What happens is, okay, do you guys remember John and Jane from our previous lectures? Yes or no? Okay, so what happens is when there's injury, uh, John and Jane, they get scared and they kind of hug each other, right? So they come together. This is the chromosome for John. This is the chromosome for Jane. They get scared. They hug each other and they clump together. This is known as chromatin clumping. Another one is if you have these sorts of changes in the cell, do you really think that the cell will have normal function, or will there be loss of function? Fast answers, please. Will there be loss of function? Yes, so there is loss of function. Okay, so there is loss of function. Another one is, as a result of swelling and mitochondrial swelling and cellular swelling and blebbing and clumping and all of these things, if there is loss of function, the protein synthesis of these patients, they will also decrease. So for NBME, you have a very important feature of reversible cell injury, and I saved the best one for the last. That is, you have, do not forget this, NBME will use this one to indicate reversible cell injury, ribosomal detachment. The ribosomes will get detached from the endoplasmic reticulums. If you have ribosomal detachment, you will have decreased protein synthesis. Okay, ribosomal detachment. Okay, so these are the 5 features of reversible cell injury. So what's happening step by step? Injury, reversible, irreversible. Reversible is characterized by cellular swelling, mitochondrial swelling. Why? Because of, always ask the question why. Why? Because sodium potassium ATPase pump is not working. As a result, osmotic imbalance, water comes inside, swells up the cells and the organelles. Since mitochondria is hampered, you have decreased ATP. Next one is loss of plasma membrane laxity results in increase in laxity of the plasma membrane result in cellular blebbing, that is protrusions of cytoplasms. Next one is chromosome clumping, okay, because this is an injury. As a result, the chromosomes kind of clump together so that there is presence of future genetic material to regenerate if needed. So chromosome clumping is basically a protective mechanism. So basically, the mnemonic is John and Jane getting scared and coming together and hugging each other in times of injury. Next one is, for all of these things, obviously there will be loss of function. And another high yield thing is that there is ribosomal detachment from the endoplasmic reticulum. As a result, you have decreased protein synthesis. These are the features of reversible cell injury. I'll give you guys 1 minute, okay? I'll give you guys 1 minute. After the 1 minute, one student will, my apologies, one physician will be confident enough to mention all the 5 properties of reversible cell injury, or else I will not move forward, okay? So I'll give you guys exactly 1 minute. We're not gonna go the same way with microbiology as we did, that is give you guys homework. We're not gonna do that anymore. for pathology, we're going to learn it right away, like how we used to. The reason being is because we would expect you guys to master first aid during our lecture, because the rest of the day should be done doing UL questions nowadays from now on, okay? So there are 5 features of cellular injury, which I want you guys to master right now. And then I'll move on to irreversible cell injury. It's 10:45 as of right now. I'll ask you the question at 10:46. Okay. Whoever is confident, please say I'm ready, and I'll ask you guys the 5 properties. Okay, Dr. Hassan, Dr. Karbasi, Dr. Hassan, you guys are all ready. Okay, who else is ready? It's 10:46. Dr. Fagan is also ready. Okay, so Dr. Jordan is also ready. Okay, perfect. Okay, so who do I ask at random? Okay, let me ask Dr. Fagan, because we have asked Dr. Hussam, Dr. Karbasi, Dr. Hassan a lot of times in the past, and also Dr. Jordan. Let's ask Dr. Fagan. Dr. Fagan, would you be kind enough to name the 5 properties of reversible cell injury?
Reversible cell, and can you hear me? Hello?
Yes, my apologies. The 5 properties of reversible cell injury.
The 5 properties of reversible cell injury, you have cellular swelling,
Cellular swelling.
blubbing of the membrane,
Okay.
loss of function, chromatin clumping,
Right.
and decreased protein synthesis.
Due to?
Decreased protein synthesis due to ribosomal detachment.
Very good. And there's also mitochondrial swelling, am I correct?
Yes, mitochondrial swelling.
And for that, we're also going to look out for decrease of ATP.
Decreased ATP, which alters the activity of the sodium potassium ATPase pumps and causes disruption in the osmotic gradient.
Right. The thing is, you have cellular swelling and mitochondrial swelling. The cellular swelling is happening because of sodium potassium ATPase pump dysfunction. And since there is also mitochondrial swelling, because let's face it, if there's swelling inside the cell, the mitochondria will be swollen too. So as a result of mitochondrial swelling, you also have decreased ATP because there's loss of electron transport chains. Is that clear, Dr. Fagan?
Yes, it is.
Perfect. Okay, so let's move on. So thank you so much. The next part, I'll ask this one to the next student after I've discussed it. Now, let's talk about irreversible cell injury. Okay. First and foremost, irreversible cell injury, the questions that you will be facing with irreversible cell injury is always remember, okay, this is high yield, what I'm going to say right now. Chromosome clumping, clumping of chromatins or chromosome clumping. This is important for reversible cell injury. And if you have nuclear breakdown, what do we mean by nuclear breakdown? Basically, if there is irreversible cell injury, the processes are basically you have necrosis and apoptosis. Let's talk about necrosis. For example, if you have long-term cell damage and the cells do not have the properties to regenerate back, the nucleus, initially they would clump together to prevent themselves from further damage. But right now, the nucleus, what they would do is they would undergo degradation. So there are 3 processes of nuclear degradation. We are talking about irreversible cell injury, okay? We are talking about irreversible cell injury. There are 3 steps of nuclear degradation. So we are talking about nuclear degradation. What happens is the step number 1, I'm pretty sure you guys know this and you guys are saying it out loud in your mind with me. It is pycnosis. Okay, what do we mean by pycnosis? Pycnosis is basically the nucleus, they get condensed together. Okay, there is condensations of the nucleus. So after pycnosis, there is Karyorexis, right? Karyorexis. Karyorexis is nuclear condensation followed by nuclear fragmentation. Nucleus, they fragment into small pieces. And then after there is fragmentation, there has to be dissolution of the fragmented material. So the last process is karyolysis. Karyolysis. If they ever mention any one of these stages in a question, please understand they are talking about irreversible cellular injury. Pycnosis, karyorexis, karyolysis, these are not the signs of reversible cell injury. These are the signs of irreversible cell injury. So when they mention this, immediately understand that the cells are undergoing irreversible cell injury. And also as a future pathologist, if you become one, then if you see these signs, then you can assume that your sample is undergoing, your patient has irreversible cell injury. That's that. Along with this, the second thing that you have to know is what's going on with the mitochondria. If there's mitochondrial swelling, then there's loss of ATP. But also over here, if you see that the mitochondria is dysfunction, if there's a damage or if there's a dysfunction to the mitochondria, then that is also a sign of irreversible cell injury. Of course, dysfunctional mitochondria would be characterized with loss of ATP and electron transport chain. So that is that. Then the next one is over here, the next one is the plasma membrane. Previously, it lost its sodium potassium ATPase pump, but right now, with continued injury, what would happen is that the plasma membrane altogether would have small breaks in them. Okay, there will be small breaks in the plasma membrane. If there's small breaks in the plasma membrane, the cytoplasm will leak out. Okay, the cytoplasm will basically leak out, and what would happen is that there will be influx of calcium. Okay, so cytoplasmic contents are leaking out, and calcium enters the cell from the extracellular environment to the intracellular environment. Whenever calcium enters the cell, always remember calcium is a potent activator of enzymes. So any enzymes that is there will get severely activated. So over here, what happens is whenever calcium enters, calcium activates the autolytic enzymes. So activations of autolytic enzymes. Okay, and activation of autolytic enzyme will result in cellular autolysis. Autolysis is basically the cells will eat itself up, the cell will get damaged, the cell will get broken down. These are the 4 steps or 4 properties of irreversible cell injury. So with that being said, we are done with the steps of irreversible cell injury. Does anyone have any question? Does anyone have any question? What do you mean by bleeding? Bleeding is basically protrusions of cytoplasmic contents. For example, if this is a cell, the plasma membrane is lax, the cytoplasm can form these kind of blebs. This is known as cellular blebbing. It should be like this, right? It should be a full round structure, but there is protrusions of cytoplasmic contents. This is known as cellular blebbing. Any other question? Part 3 of irreversible. Part 3 of irreversible is there is breaking down of plasma membrane, leakage of cytoplasmic fluid, and increased entry of calcium from the extracellular environment. When calcium enters the cell, it activates the autolytic enzymes, and there is autolysis. Okay, now I'll give you guys 1 more minute, and then whoever is confident, please say I'm ready, and I'll ask you guys, and then I'll move on. One minute on the clock. Whoever is ready, please say I'm ready. I'll ask you the question and I'll move on. Yeah, so I'll show the book. Don't worry. First, just try to remember these four properties. Okay, Dr. Karbasi is ready. Who else is ready? Okay, Dr. Hussam, Dr. Karbasi. Okay, so Dr. Karbasi, what are the four properties of irreversible cell injury?
It's like nuclear degradation, which is called pyknosis, which is nuclear condensation, chirorexis, nuclear fragmentation, and chemolysis. Then we have the mitochondrial dysfunction due
Okay,
to which leads to decreased ATP due to loss of electron transport change.
very good.
Third one is like we have like a damage in the cytoplasmic membrane, which then leads to leakage of cytoplasmic fluid, which then leads to influx of calcium, which then
Okay,
activates autolytic hemolytic enzymes, which then cause autolysis, which the cells eats themselves, and we have a cell injury, right?
very good. So these are the properties of irreversible cell injuries. Okay, so let's see. We have breakdown of plasma membranes, mitochondrial dysfunction, cytoplasmic vacuolation, accompanies programmed cell death. We'll talk about this, cytoplasmic vacuolization, rupture of lysosomes and autolysis, nuclear degradation, and amorphous densities or inclusions in the mitochondria. So basically, you have mitochondrial inclusions or mitochondrial swelling. But this is not very high yield, to be honest, because you do not really have, you do not really see a lot of mitochondrial inclusion bodies. So the fact that there is mitochondrial inclusions, this can happen in either cases. So this is not very characteristic of irreversible cell injury. The outmost important ones are these ones, um, one, two, three, four. Okay, these are the four ones. So if you have to remember them, please try to remember these four. If you can remember extra information, that is mitochondrial inclusion and cytoplasmic vacuolation, that's good on you. Okay, so that's that. With that, before we move forward, let's look at the picture. Okay, this is cellular blebbing, as you can see, cytoplasmic protrusions, nuclear clumping, ribosomal detachment, and cellular swelling. So these are the properties of reversible. Irreversible is characterized by rupture of the enzymes, nuclear degradations, mitochondrial permeability, and mitochondrial swelling, loss of ATP, along with plasma membrane damage. So the cells are dying, cell death. Okay, now let's move on to apoptosis. Okay, are we ready to begin our discussion on apoptosis? Everyone, are we ready to begin our discussion on apoptosis? Does anyone have any questions from cell injury? Okay, so apoptosis is basically, okay, apoptosis is basically, it's an ATP dependent programmed cell death. So this line over here, okay, this line over here, ATP dependent programmed cell death, they use these phrases, they use the phrase programmed cell death to indicate that the process is apoptosis. Okay, programmed cell death for apoptosis. There are two types of pathways, intrinsic pathway and extrinsic pathway, and we'll break this down because this is a very high yield diagram. We'll break this down in 1 second. Let's read this first. There's intrinsic and extrinsic pathways. Both pathways will activate caspases. Caspases are basically cytosolic proteases that are released from the mitochondria. So there's cellular breakdown, including cellular shrinkage, chromatin condensations, membrane blebbing, and formations of apoptotic bodies. My question to you guys is, if we have apoptosis, does that mean the cell is undergoing reversible or irreversible injury? Fast answers, please. The cell is undergoing irreversible injury. If there is apoptosis or if there is necrosis, then the cell have undergone irreversible injury. That's why when you stain the cell with eosinophilic cytoplasm, and I mean, when you stain the cell, you will see that there is a eosinophilic cytoplasm and a basophilic nucleus, meaning that there will be a pinkish cytoplasm and a bluish tinge to the nucleus, showing that there is nuclear condensation, nuclear fragmentation, and nuclear dissolution, meaning pycnosis, karyorrhexis, and karyolysis. The cell membrane typically remains intact without significant inflammation, unlike necrosis. So what happens is you either have blebbing or you either have breakdown. In necrosis, there is full-on breakdown of the plasma membrane. In apoptosis, you have significant inflammation, which results in laxity of the plasma membrane. So there is no breakdown of the cell, there is blebbings only, okay? So this is a very major difference between necrosis and apoptosis, is that in necrosis, you will have no blebbing. In apoptosis, you will have blebbing, okay? That's that. And necrosis is not a programmed cell death. It's an unprogrammed death, and apoptosis is a programmed cell death. There's a DNA laddering, meaning that fragments in multiples of 180 BP is a sensitive indicator of apoptosis. Basically, if we mean that fragments, meaning that the nucleus is broken down into small fragments, and if the fragmentations of the nucleus are more than the multiples of 180 BP, then this is a sensitive indicator of apoptosis. This is not high yield. You do not have to remember this, okay? This does not have to be remembered, okay? Now, while my page on Cami pops up, does anyone want to tell me the properties of irreversible cell injury one more time, please? What are the properties of irreversible cell injury? Anyone? One more time. Properties of irreversible cell injury. Okay, Dr. Hossam, let's hear you say it.
Yes, we have, you mean apoptosis, yes?
Yes.
Apoptosis.
Yeah. No,
okay.
Irreversible cell injury.
Okay, degradation of cell membrane,
okay,
and increased permeability of mitochondria,
okay,
and accumulation of autophagic lysosome. Or
what is the first thing you will see
clump,
in an irreversible cell injury?
nucleus, karyorrhexis, and pycnosis.
First step is pycnosis, right?
Yes.
Nuclear condensation followed by nuclear fragmentation. Then there is nuclear dissolution. So pycnosis, karyorrhexis, then there is karyolysis. That's the first step. Next one you said was cellular breakdown and mitochondrial dysfunction. That is okay. Then
yes.
what is the next one?
Lysosomal rupture.
Nope, lysosomal rupture is not the one. That is, you have calcium influx. Do you remember calcium influx?
Yes. It's
Calcium
due to
influx
membrane damage, yeah?
Yes, membrane damage, calcium influx. When calcium enters, it activates which enzymes?
Lysosomal enzymes.
Very good. It activates the lysosomal enzyme. That as a result, you have autolysis. Are we clear about this?
Yes.
Okay. Now let's talk about apoptosis. So we talked about apoptosis over there. Now the apoptotic pathway, we have to understand how this programmed cell death is happening. So apoptosis, if we have to talk about this, apoptosis, okay? Apoptosis basically have two pathways. One is intrinsic pathway, meaning that this is the pathway of the mitochondria. Another one is extrinsic pathway. Okay, intrinsic and extrinsic pathway. Okay, please excuse my handwriting; it's absolutely horrible. Now let's start talking about the intrinsic pathway at first. Okay, so if we have to draw a cell, okay, let's draw a cell first. Okay, what's happening over here? Now this cell will undergo apoptosis. So intrinsic pathway is happening. For example, there are two reasons. Okay, for example, there are two reasons of intrinsic activation of intrinsic pathway. Activation of intrinsic pathway of apoptosis, number one cause is if there is loss of cellular regulation. Okay, if there is loss of cellular regulation, or if there is increased injury or insult to the cell. increased injury or insult to the cell. Increased injury or insult to the cell, this could be by toxins, this could be by drugs, this could be by hypoxia, this could be by prolonged radiation, so increased injury, or if there's loss of cellular regulation. For example, if you take away a regulating factor, for example, we know that, let's take the example of T cells, okay? Which interleukin is responsible for the growth and differentiations of T cells, for growth and differentiations of T cells? Most of the T cells. Which interleukin is responsible? Fast answers, please. Very good. Interleukin 2. Okay. Interleukin 2 is there. If you do not have interleukin 2 stimulation, will there be growth of the T cells? No. Okay. So loss of interleukin 2 is an example of loss of cellular regulation. Now, I want you to put your attention over here. Okay. I want you to put your attention over here. Now, what happens is you have these damages. Okay. You have these stimulators. So let me write this down. Okay, so you have stimulators. The stimulators are these ones. These are the stimulators. So they will stimulate intrinsic pathway. So these stimulators, what they would do is, these stimulators, they will activate a very important tumor suppressor gene, that is P53. So activation, they will activate P53. P53 in turn will activate two types of proteins. Okay, two types of proteins. These proteins, they basically belong to the BCL family. These proteins are known as BAX. Okay, these proteins, one of them is known as BAX, another one is known as BAC. Okay, another one is known as BAC. So once again, these two are the stimuli. They will activate P53. P53 will activate BAX and BAC. BAX and BAC, they they belong to the BCL family. BCL, the one that is, for example, in which type of carcinoma is BCL there? BCL, who remembers BCL? Very good. We have BCL in the follicular. By follicular, Dr. Hussam means follicular lymphoma. So it belongs to the BCL family. So BCL family have BAX and BAC. So what BAX and BAC, what they would do is they would stimulate, okay, they would stimulate the mitochondria to secrete a very high yield structure. This structure is known as cytochrome C. Okay, I'm going a bit slow so that you guys don't lose the track. So once again, there's stimuli. They will activate P53. P53 will activate BAX and BAC. BAX and BAC will activate macrophage. Macrophage will stimulate the release of cytochrome C. Cytochrome C, when they get activated, cytochrome C will activate initiator caspase. Okay, caspase enzymes. Okay, caspase enzymes are cytolytic enzymes. Cytolytic enzymes mean they will break down the cytoplasm. Okay, so cytolytic enzymes. Caspases will activate, initiator caspase will activate execution, meaning that there are two types of caspase. One will initiate the process of apoptosis, another one will execute the process of apoptosis. So there are executory caspases. Executory caspases will do two things. First of all, they will break down the cellular cytoskeleton. Okay, first of all, they will break down the cellular cytoskeleton. So cytoskeleton damage. Another one is, what they will do is they will break down the nucleus. So the nuclear pycnosis, karyorrhexis, karolysis, this is what you will see. So nuclear damage. So cytoskeleton damage and nuclear damage. These cytoskeleton damage and nuclear damage, what they would do is they would cause the creation of these sorts of cytoplasmic apoptotic bodies, or blebs. Okay, they would cause the formations of these types of blebs. These blebs would contain the cytoskeletal fragments. They will contain the nuclear fragments. Okay, and along with this, they will have these types of receptors over here. Okay, these types of receptors over here. These receptors, they will go and activate macrophage, and macrophage will come and cause phagocytosis. Okay, macrophage will come and cause phagocytosis. This is the process of intrinsic apoptotic pathway. Are we all clear? Yes or no? Are we all clear about this? Yes or no? Okay, I'm going to repeat this. First of all, apoptosis is a irreversible cell injury. Irreversible cell injuries have apoptosis. Irreversible cell injuries have necrosis. Apoptosis have two pathways: intrinsic pathway and extrinsic pathway. Intrinsic pathways are induced by loss of cellular regulation. For example, loss of interleukin 2 for T cell growth and proliferation. That's an example. Another one is if there is injury by cellular toxin, drugs, radiation, hypoxia, loss of blood. So these are the stimulators. So when you have these two things happening, the intrinsic pathways will get stimulated. This will activate P53 enzyme, P53 gene. P53 is a tumor suppressor gene. P53 activation will activate backs and back of the BCL family. They will activate the mitochondria to release cytochrome C. Cytochrome C will activate initiator caspase. Initiator caspase will activate executionary caspase. Executionary caspase will break down the cytoskeleton and they will cause nuclear fragmentation and damage. And this will result in laxity of the plasma membrane and depositions of the fragments in this sorts of blebs, which will activate the macrophage, and the macrophage will start phagocyting the cells. Okay, this is the process of intrinsic apoptotic pathway. Is everyone clear? Yes? Okay. Now let's read. Okay, let's read this. Intrinsic pathway, it's involved in tissue remodeling in embryogenesis. So basically, the process they're talking about over here is physiological apoptosis. For example, during embryogenesis, a lot of the structures undergo apoptosis, right? To give rise to a lot of matured structures. So there is physiological apoptosis, and there's in pathological apoptosis. This occurs. Excuse me.
I have a question.
You have a question? Okay, just give me 1 minute. Let me just
Okay.
get done with the thing, and then I'll ask you the question if you see it. If you have any. Okay. Occurs. So where were we? We were in intrinsic pathway. Okay. So it's involved in tissue remodeling and embryogenesis. It occurs when a regulating factor is withdrawn and proliferating from a proliferating cellular population. That is, if you have interleukin 2 as an example, which stimulates the growth and differentiations of T cells. If you take it away, then there will be no future growth and differentiations of T cells, and as a result, there will be cellular apoptosis. That's one. Another one is when they're exposed to injurious stimuli, for example, radiation, toxins, hypoxia, the ones we just talked about over here. Now, these will activate the regulators of BCL2 family of proteins, the backs and back. Please remember, backs and back they belong to the BCL2, while BCL2 and BCLX are anti-apoptotic. So over here, you have two processes going on. We just talked about the apoptotic process. We did not talk about the anti-apoptotic process. That is, backs and back will stimulate the mitochondria to release cytochrome C, but in the other hand, BCL2 and BCLXL can inhibit the mitochondria to release cytochrome C. So basically, this is like a positive negative feedback kind of a situation going on. Okay. Backs and back, they form pores in the mitochondrial membrane that will release the cytochrome C. So they form pores from the inner mitochondrial membrane into the cytoplasm. And this results in the activation of initiator caspase and where were we? The activation of initiating caspase. And the BCL2 keeps the mitochondrial membrane impermeable, thereby preventing cytochrome C release. So backs and back will promote the release of cytochrome C once again, and BCL2 will inhibit the release of cytochrome C. Once the cytochrome C is released, then it's game over, because the cytochrome C will activate the initiator caspase. Initiator caspase once again will activate the executionary caspase, and then there's cytoskeletal degradation, nuclear degradation, apoptotic body, and phagocytosis. Okay, this is the example. For example, if there is BCL2 overexpression, BCL2, what we have just learned that it's an anti-apoptotic. That's why if there's high amount of BCL2, some cells, they do not undergo apoptosis. As a result, they can undergo unregulated proliferation and uncontrolled growth, giving rise to carcinoma. For example, follicular lymphoma is a type of this example, T1418, which occurs due to decreased caspase activation and increased tumor genesis, meaning that since there is no apoptosis, the cells keep on growing and growing and growing, and then at one point there's unregulated proliferation and resulting in carcinoma, that is tumorogenesis. Now, we are done with the discussion of intrinsic pathway. Okay, so who was the physician with the question? Who unmuted
Here,
you? Yes, what is your question, please?
the question is, P53 also plays a role in DNA repair. And how does the cell differentiate between repair and apoptosis?
Okay, good question. So basically, your question is P53 has an role to play in DNA repair. And over here, how does, okay, good. The thing is basically P53, one is loss of P53, another one is there is activation of P53. So P53 is a tumor suppressor gene. Basically, if you want to suppress a growth of a cell, you will activate P53 because P53 activation will result in backs and back. If you want to have DNA repair, if DNA has to be repaired, I'm not sure how much of P53 activation happens over there, if that is a very important step in the process of DNA repair. But having said that, we cannot exclude the importance of P53 in DNA repair. So what happens is that in DNA repair, if there is activation of P53, then P53 activation will again induce apoptosis. So in the process of repair, P53 has to be silenced. So if there's silencing of P53, then what happens is there is no activation of backs and back, and the cell can repair easily. Okay. Does that help answer your question?
Yes, thank you.
You're welcome. Does anyone have any other question? Yes or no, guys, please. Yes or no? Yes for questions, no for no questions. Okay, good. Perfect. No questions. If there are no questions, I'll give you guys 1 minute once again. And whoever is confident enough can please explain the pathway of intrinsic apoptosis before we move on to extrinsic. One minute on the clock. It's 11:18. I'll ask you the question at 11:19. Dr. Krabasi is ready. Very good. Who else is ready? Okay, thank you, Dr. Krabasi, for putting your attention to the lecture. Everyone else, thank you to you too. Please try to participate in active participation. Dr. Hassan is ready. Thank you. Is there anyone else who is ready? Because we already asked Dr. Krabasi, we already asked Dr. Hassan. Who else is ready? It's not a very difficult step, okay? I'm just gonna tell you guys a step and exactly tell you what happens. Basically, you have DNA damage. DNA damage will activate P53. P53 will activate backs and back. Backs and back will activate the release of cytochrome C. Cytochrome C will activate initiator caspase. Initiator caspase will activate executionary caspase, and that will activate nuclear fragmentation and cytoskeleton degradation, followed by the formation of apoptotic bodies and macrophagic phagocytosis. That is what I want to hear. Dr. Mridula is ready. Okay, so let's hear it from Dr. Mridula. Would you be kind enough to tell us the pathway of intrinsic apoptotic pathway?
Can you hear me?
Yes, I can hear you. What are the pathways?
The two pathways, intrinsic and extrinsic. In the intrinsic pathway, the stimulators are loss of cell regulation, which is reduction of IL-2, and also increased exposure to the injurious stimuli.
Okay.
After that activates P53, which in turn activates backs and back.
Okay.
That stimulate mitochondria to release cytochrome C.
Okay.
Cytochrome C activates initiator caspases,
Okay.
which activates executory caspases.
Okay.
That causes breakdown of cytoskeleton and nuclear damage, which results in formation of apoptotic blebs that
Okay.
activate macrophages,
Okay.
and macrophages cause phagocytosis.
Perfect. Okay, perfect. That was the exact process. Thank you, Dr. Midula. And another thing is, interleukin-2 is just an example. It's not an important step of apoptosis. So this is just an example. Okay, so this is an example of loss of cellular regulation. So not for every time apoptosis to take place, interleukin-2 does not have to decrease. Okay, so that's that. So intrinsic pathway is done. Extrinsic pathway is way easier than intrinsic pathway. Okay, extrinsic pathway, what happens is that in extrinsic pathway, there are two things that's happening over here. Okay, let's talk about extrinsic pathway. I'm going to try to use the same thing. The thing is, in extrinsic pathway, you have two things that can cause the damage. Okay, so number one is FAS-L, active FAS-L interaction. Okay, FAS-L interaction or tumor necrosis factor interaction, TNF factor interaction. And another one over here is cytotoxic T cell interaction with the cell. So basically, if you can try to understand, intrinsic pathways are happening because of cellular damage inside the cell. Extrinsic apoptotic pathway is being activated because extrinsic cells are causing the damage to the cell themselves. It's not because of the cell is being exposed to any injurious stimuli. It's because outside the cell there are some other cells which wants this cell to die. So that's the thing. Now, if I want to talk about the first process, that is FAS-L and or tumor necrosis factor causing the damage. So first of all, I'm pretty sure we are all smart enough to understand that in order for interactions to take place, there has to be receptors. Okay, so let's assume that this is the FAS-L receptor, and let's assume that this is the TNF receptor. So basically, if this is a FAS receptor and FAS-L will come, okay? So FAS-L will come, 1 second. FAS-L will come and bind to this receptor, or tumor necrosis factor will come to bind to tumor necrosis factor receptor. It's not important for both of them to come and bind at the same time to activate the pathway. Either one can come, either one, either or, either FAS-L or either TNF, or both of them can come together. It's their choice. So they can come, and what happens is, whenever there is interaction of FAS with FAS-L or tumor necrosis factor with TNF receptor, there is activation of initiator caspase directly. There is direct activation of initiator caspase. When there is direct activation of initiator caspase, once again, initiator caspase will activate executionary caspase. Executionary caspase can stimulate the formation of apoptotic bleb, and there could be macrophagic phagocytosis. Try to understand that in the process of extrinsic pathway, cytochrome C is not important, because mitochondrial release of cytochrome C is only important for intrinsic pathway, and this is high yield for NBME. Which process involves cytochrome C? The answer is intrinsic pathway. In extrinsic pathway, they directly activate the intrinsic, the initiator caspase, which causes the cellular damage. So this is one pathway. This is one way. Another way is if there's a cytotoxic T cell. If there's a cytotoxic T cells, what they do is, do you guys remember we talked about this in the past, that cytotoxic T cells, they release two types of enzymes, granzymes and perforins. Okay, who remembers this? Let's see. You guys remember granzymes and perforins released by cytochrome C? Yes. I mean, by cytotoxic T cells. So cytotoxic T cells, whenever they come close together, what they do is they causes the release of, for example, let's say that this is the This is the granzyme that has been released by this cytotoxic T cell. So the granzymes, my apologies. This is the perforin. Perforins are the one that will perforate the cell membrane. So for example, let's say this is the cytotoxic T cell. This is the cytotoxic T cell that came in close contact to this cell. And this cytotoxic T cell released perforin, which perforated the plasma membrane of the cell. And after perforating, the cytotoxic T cell released its granzymes. Okay, granzymes. Granzymes, what they would do is, granzymes, instead of activating initiator caspase, granzymes would directly activate the executionary caspase. Granzymes would directly activate the executionary caspase, and that will cause the cytoskeleton damage, nuclear fragmentation, apoptotic bodies, and macrophagic phagocytosis. So these are the 2 steps of extrinsic pathway. Now, let's look at this. Extrinsic pathways have 2 pathways. One of them is ligand-receptor interaction, that is FASL. FASL is also known as, the FAST is also known as cluster differentiation 95 or CD95, or TNF-alpha binding to its receptor. Or immune cell, for example, cytotoxic T cell release of perforins and granzymes. So if there's a FAST-FASL interaction, it is necessary in the FAST-FASL interaction is necessary in the thymic medullary negative selection. Do you guys remember when we talked about negative selection? What happened in negative selection? Did the immature T cells bind to the body cell, or did they not bind to the body cell? Negative selection. What happens in negative selection? First of all, in negative selection, not bind or bind? Bind. Okay. In negative selection, if the T cells bind to the body's self antigen, then that T cells can undergo autoimmunity in the future. So T cells who binds very strongly with the body's own cell, they will undergo apoptosis, and FAST-FASL interaction is necessary to induce apoptosis in those T cells. So FAST-FASL interaction is necessary for negative selection. Guys, this is the question. Which process of apoptosis is responsible for negative selections in the thymic medulla? This question, the answer is FAST-FASL interaction, okay? Do not forget this. No other one. TNF is not there. Cytotoxic T cell, not FAST-FASL is there, okay? Fast mutations. So obviously right now, if we have fast mutations, can we assume that there is a high amount of autoimmune T cells? Yes. Because in negative selection is not happening. So that's what's here. Fast mutations is increased number of circulating self-reactive T cells due to failure of clonal deletions. Defective FAST-FASL interactions can cause autoimmune lymphoproliferative syndrome, not high yield. Don't worry about this. Just understand that if there is damage of FAST-FASL, then there will be autoimmunity. Autoimmunity is due to one of the many, many reasons of autoimmunity is FAST-FASL interaction abnormality. Okay. So with that being said, we are done with the extrinsic pathway of the apoptosis. I'll give you guys 1 minute once again. Okay, before I give you guys a minute, does anyone have any question? Does anyone have any question? Okay, no question. So I'll give you guys 1 more minute.
Can you explain the negative binding and the
Which one?
The negative selection and it being destroyed?
Oh, negative selection is basically when this is the process for T cell growth and maturation. So first of all, when there's a dendritic cell which takes an antigen up to the thymus, so at first there's positive selection where Yeah, I'm going to try to explain this into a brief manner because it's a big discussion. So first process is positive selection, where the T cells are tested to see if they can bind to the body's own antigen. So the T cells which can bind to the body's own antigen, they are selected. Then the next step is negative selection. That is, they want to see if they do bind to the body's self antigen, how strongly do they bind. If they bind very strongly, then the body realizes that there is a possibility that these T cells will be autoimmune T cells in the future. So the body, what it does is it induces the apoptosis of those T cells in the thymic medulla. This process is known as negative selection. And the process of negative selection and clonal deletions of the thymic T cells in the medulla happens by the process of Fas-L and Fas interactions. So we can assume that if there is no Fas-L interaction between the T cells, then there is a possibility that the T cells will survive and cause autoimmunity in the future. So that's what we were discussing over here. Hope that answers your question. Does that answer your question?
It's complicated, but it makes a lot more sense. So if that
it would make a lot more sense if did you study immunology yet?
No, I haven't.
Okay, so when we study immunology in the future, I'll break it down obviously into more easier pattern, but just try to understand for now that T cells have to be activated. For T cell activation, there's a positive selection where we introduce the T cell to the body's own antigen so that it doesn't so that it understands who to attack and who not to attack. It's basically, for example, let's say if there are soldiers of the country, the soldiers go around in the country trying to look for the people of the country. And for example, if they see the soldiers being attached to a single person way too seriously, then that soldier has to be taken down. It's something like that of that nature. Basically, if you have a positive selection, the T cells recognize the body's own antigen at first, and in the negative selection, we want to see how strongly they attach themselves to the body's own antigen. If they attach too strongly, then the body thinks that there's a possibility for these T cells to be autoimmune, so they cause Fas-Fas interactions and damage to the T cells and makes the T cell die. This is known as clonal deletion. Okay. Does that help a little bit?
Thank you. Yep.
You're welcome. We'll go into the details of this when we study immunology. Having said that, who is ready to tell me the steps of extrinsic pathway? Very easy, guys, come on. There are two steps: ligand receptor binding. Another one is T cell. Ligand receptor has Fas-cell interaction. Another one is cytotoxic T cell. Both of them will activate the caspases. Fas-cell will activate initiator caspase, and that will activate executionary caspase. And TNF would also do the same. Cytotoxic T cells, on the other hand, would release perforins and granzymes that will activate executionary caspase directly and cause damage. As simple as that. Who is ready? Dr. Fagan is ready. Okay, who else is ready? Dr. Karbasi is ready. Okay, Dr. Fagan, Dr. Karbasi, Dr. Hussam, thank you so much for trying to participate. Dr. Mudula is also ready. We asked you guys. Well, I would really appreciate if anyone else would also try to participate, please. Who else? Okay, so let's not waste any more time. Dr. Fagan, would you be kind enough to once again answer the extrinsic pathway patterns?
Okay, the extrinsic pathway is stimulated by like two signals. You have the binding of the Fas ligand
Right.
or the TNF to the cell receptor,
Right.
which is going to cause direct activation of the initiator capsases.
Okay. They will directly activate the initiator caspases. That's correct then.
And then that will initiate the executory capsases, which will cause breakdown of the cytoskeleton and the nucleus.
Good, very good. So that is ligand interaction. Next one is which another one did you say was also responsible for activating the execution?
The second one is the cytotoxic T cells, which cause direct release of perforin and granzyme, and that will just directly activate the executory
Executionary, you're right. That will directly activate the executionary caspases, and then there will be the same process.
Okay.
Perfect. So that is exactly what it is. Thank you, Dr. Fagan, for answering the question. Now, let's move on to the next one, that is necrosis. Okay. Necrosis. Now, please understand, the most important thing about necrosis is histology. Okay. Let's start talking about necrosis. So we're done with apoptosis. Necrosis is exogenous injury, meaning that there is plasma membrane damage. Cell undergoes enzymatic degradation and protein denaturation, and intracellular components will leak. Very simply put, apoptosis is not characterized by, apoptosis is not characterized by breakdown of cell membrane, but necrosis is characterized by breakdown of cell membrane and leakage of cytoplasmic contents. And leakage of cytoplasmic contents can induce a local inflammatory response. Okay, so once again, apoptosis, no breakdown of plasma membrane, necrosis, breakdown of plasma membrane. Now, how many types of necrosis there are? There are six types. Okay, number one is coagulative necrosis. Okay, the easiest way to identify coagulative necrosis is always remember coagulative necrosis will happen in compact organs. What do I mean by compact organs? For example, if there's a necrosis that happens in the heart, the type of necrosis that will happen is coagulative necrosis, because the organ, the heart, is a very compact organ. Okay, so it will happen in most tissues. For example, the most common ones are once again heart. Then we have the kidneys, right? Because they are very compact organs. So over there you can assume to see coagulative necrosis. So they are seen in most tissues except brain, because brain is not a compact organ. Over there are spaces in between the brain, which can result in a different type of necrosis. I'll talk about that later. Now, the reason of coagulative necrosis is infarction. For example, in the heart, you have, if there is atherosclerosis, loss of blood supply, this can induce an infarction. Injury that can denature the enzyme, and enzyme denaturation can result in proteolysis block. Okay, so that's that. So over here, you have to focus on ischemia infarcts in most tissues, but I believe this is not something which you guys should learn. This is something which you guys should know already. Okay, so I'm not going to put a lot of focus on this. I want to put all my attention to this one over here. That is the histology. That is, in coagulative necrosis, we have to understand that the cellular architecture is preserved. As a matter of fact, over here, as you can see, that the cell lines can be seen. That the cellular architecture over here is more or less preserved, but that there is disappearance of the nucleus. Can anyone tell me why is there disappearance of the nucleus? Fast answers, please. Can anyone tell me why there is pycnosis? Very good. Pycnosis, then which one? What happens after pycnosis? Karyorrhexis and karyolysis. They're very good. Because necrosis is an irreversible cell injury. Okay, so because necrosis is an irreversible cell injury. And over here, you will see increased cytoplasmic binding of leucine stain. That is increased eosinophilia. That is, there will be increase of red colored of the same. Okay. Okay. That's that. Next one, liquefactive necrosis. Okay. Liquefactive necrosis is, okay. Do you guys want me to talk about this in the blank page, or do you guys want me to use the first aid? I'm using this because to save some time. Which one do you guys prefer? Because I will not move on until and unless. Okay, first aid. Okay, good. Liquefactive necrosis. Now, let's go. So let me at least tell you which ones you have to remember. That is cellular architecture preservation and nuclear disappearance is high yield. Okay. Liquefactive necrosis. Liquefactive necrosis happens in most in organs which have spaces between them. For example, brain is an excellent example of liquefactive necrosis. And for example, if there's infarction in the brain, then there could be liquefactive necrosis where the neutrophils will release lysosomal enzymes that digest the tissue. Okay. Okay. if there is an organ with spaces in between them that is not a compact organ, does it not give more chance for the calcium to enter the different types of cells? Yes or no? If the organs are not compact, because there is a more extracellular space in between the cells. Yes. Okay. So if you can imagine that organs which have a bit more spaces in between them, for example, brain is an example, this allows for more calcium to enter the cell from the extracellular environment into the intracellular environment, resulting in activations of the enzymes for lysosomal degradation. So always remember, liquefactive necrosis happens in organs which have a bit more space in between the cells, and brain is an excellent example. In early stages, you will see central debris and macrophage. In late stages, you will see cystic spaces and cavitations. Neutrophils and cellular debris is seen with bacterial infection. Basically, the cell that you have to focus on is neutrophils. If you can see polymorphonuclear cells, and over there also with the fact that don't forget that you would also see pycnosis, keratosis, that is loss of cellular nucleus, right? And in the early stage and late stage, basically the questions will not come under the way for you to differentiate if this is an early stage or if this is a late stage or not. The questions will come for liquefactive necrosis, and you will easily identify these questions only by their location, because USMLE step 1 will only use brain for necrosis. Whenever they say brain and necrosis together, please remember they're talking about liquefactive necrosis. And also, to help you out a bit more, if they have a picture, you will see that there is polymorphonuclear cells, okay, and there are cystic spaces and cavitations. That is that. Next one, caseous necrosis. Caseous necrosis, okay, very high yield, I mean, very common for tuberculosis, okay. Sarcoidosis, do we have caseous necrosis in sarcoidosis? Yes or no? Good, very good. So tuberculosis is tuberculosis, then we have fungi, histoplasmosis, then can you guys name some more fungi that can cause caseous necrosis? Aspergillus, nope. Aspergillus causes aspergilloma, or invasive aspergillosis, or Aspergillus or allergic bronchopulmonary aspergillosis. They do not necessarily cause caseous necrosis. Which fungi cause caseous necrosis? Histoplasma is already written over here. Coccidiomycosis is a very good example. What else? Blastomycosis is a very good example. Then what else? Last one. Cryptococcus, okay, that's a very good example. Okay, good. So these are the ones. Then nocardia can also cause caseous necrosis. Okay, nocardia can cause caseous necrosis. If nocardia is causing caseous necrosis, and if it's an anaerobic organism, what is the anaerobic counterpart of filamentous bacteria? Actinomycosis. Actinomycosis, very good. Okay. Can we identify actinomycosis with sulfur granules? Okay. Are they gram positives or gram negatives? Gram positives. Very good. Okay. Now, caseous necrosis is very easy to understand because there will be a central necrosis surrounded by a wall of epithelioid cells. Epithelioid cells are basically giant macrophages. So macrophages will wall off the infected microorganism, followed by granular debris. Okay, as you can see over here that this is a lung with the tubercular granuloma. Okay, there's a central necrosis and the surrounding epithelioid macrophagic border. If you do histology over here, you can see that there's fragmental cells and debris surrounded by lymphocytes and macrophage. Basically, if you have any type of necrosis surrounded by macrophage, that is caseous necrosis, especially in the case of, for example, tuberculosis or histoplasmosis. So basically, if they make a case or if they make a question about caseous necrosis, they will never fail to mention these organisms. Tuberculosis, systemic fungi such as histoplasma, blastomycosis, coccidiomycosis, paracoccidiomycosis, then cryptococcus is also there to some extent. Okay, but these are the ones. Nocardia for bacteria. Okay, so that's that. Okay. Next one is fat necrosis. Okay. Fat necrosis is over here, there is enzymatic and non-enzymatic. And enzymatic, you have acute pancreatitis. For example, fat necrosis is basically the fat component of the cells will get broken down, or they will die, and they will be replaced by calcium deposition. So basically, if there's acute pancreatitis, what happens is after acute pancreatitis, there is calcium deposition and there's saponification, meaning that there's soap formation of the pancreatic fat. The word saponification basically means that there is soap formation by the deposition of calcium. Okay, so saponification. And non-enzymatic is, for example, if there is a patient who has recently been in a mass gathering, and for example, there was the patient did not realize, but that the patient got hit on her chest by a solid object. For example, let's say let's say a woman is running, and out of nowhere, the patient gets hit on her chest, but the hit is not that strong enough to induce a painful reaction. Okay, it's not a very strong hit, it's a very blunt hit. Boxing is there, yes, for boxing is there, but the thing is, the question for fat necrosis of the breast will come that the patient will not remember the trauma that the patient has went through. So basically, the question will tell you that when the doctor asked the patient if the patient had any trauma, the patient will not be able to recall the trauma. Okay, so the trauma will not be recalled. Okay, I remember one question from MBOS about a young girl who was involved in a mosh pit. Do you guys know what a mosh pit is? Has anyone been to any concerts in their life? No concerts. Okay. Okay. Mosh pits are basically crazy people during concerts. When they get really crazy, they start pushing each other in the middle because apparently the music is so good for them that they just want to fight or something. I really don't understand the idea of mosh pits, but that's what it is. Okay. So in mosh pits, there's this young girl who got involved in a mosh pit in a concert, and after she came back home, she realized a sort of mass on her breasts. Okay, the question will direct you towards the answer of a breast carcinoma or something like this, but the answer is actually fat necrosis. That is, when the young girl was involved in the mosh pit, of course, she got hit on her chest, but she did not realize it. And what happens is that afterwards, that fat component in her breast tissue was replaced by calcium deposition. So this is fat necrosis. So damaged pancreatic cells release lipase, which breaks down triglycerides. These are the examples of enzymatic fat. Don't confuse this with non-enzymatic. So pancreatic cells, which will release lipase, will break down the triglyceride, and fatty acid will bind to the calcium and saponification. This is what happens. So there's chalky white appearance. Over here, if you can see that there's outlines of dead fat cells without peripheral nuclei, meaning that the nucleus is not there. Of course not, because it's a necrosis. So it's an irreversible cell injury. So the nucleus is not there, but there is depositions of these types of chalky white appearance of these calciums. So this is fat necrosis. Okay, that's that. Next one is fibrinoid necrosis. This is extremely high yield. Okay, let me tell you one of the most high yield necrosis is over here. Coagulative necrosis, liquefactive necrosis is high yield, but it's easy to understand. So I'm not going to put a star. Caseous necrosis is high yield. Fat necrosis is not that high yield. Fibrinoid necrosis is very high yield. Fibrinoid necrosis is basically immune vascular reactions are an example. For example, polyarteritis nodosa, or there's fibrinoid necrosis. Non-immune vascular reactions, for example, hypertensive emergency and preeclampsia. In hypertensive emergency, you have fibrinoid necrosis, okay? And also in preeclampsia, you have fibrinoid necrosis. Basically, this is the vessel walls, they become thick and pink because of loss of fibrinoid-like material. So immune complex deposition can induce a type 3 hypersensitivity response, and plasma protein can leak. So basically, if there is a cellular damage, there is increase in space in between the adjacent cells, which allows leakage of the plasma protein. And this plasma protein kind of accumulates around the damaged cells, and they stick together, and then they get hardened. Okay, then they get hardened to form this sort of feature. This is known as fibrinoid necrosis. Okay, the vessel's wall, they become thick and pink because of the excessive leakage, and excessive leakage and accumulation of the plasma protein. So that is fibrinoid necrosis. Okay, so we're done with all the hard ones, meaning gangrenous one is the most easiest one to understand. Gangrenous necrosis is basically gangrene. There are two types of gangrene, dry gangrene and wet gangrene. Dry gangrene is basically there's ischemia without infection, and wet gangrene is there's ischemia with infection. Okay, so as you can see over here, this could be easily a patient of excessive this could be a patient of diabetes mellitus with a very poor control of their glycemic levels, resulting in peripheral neuropathy and then loss of innervation and then damage and gangrenous necrosis over here, as you can see. Okay, this is the most important causes due to ischemia. Can anyone tell me why do we have ischemia in diabetic mellitus? Why do we have ischemia in the peripheral arteries or the peripheral blood vessels? Very good, glycosylations. Okay, there is glycosylations and okay, glycosylations, very good. So glycosylations are number one. That is the arteries, they get thick and then there's decreased blood supply followed by ischemia and gangrene. If there is gangrene with no infection, it's called a dry gangrene. If there's a gangrene with infection, it's called a wet gangrene. Okay, so that's that. That is gangrenous necrosis. Now, before I move forward, I'm going to give you guys 2 minutes to tell me the histology of coagulative necrosis. I want to hear you guys say that in coagulative necrosis, we have preservations of cellular architecture, but nucleus is disappeared. Nucleus has disappeared, and the cytoplasm has a eosinophilic tinge. In liquefactive necrosis, I want to hear you guys say that there is neutrophilic accumulation and there are cystic spaces. In caseous necrosis, I want to hear you guys say that there is a central caseous necrosis followed by a rim of epithelioid cells. In fat necrosis, I want to hear you guys say that there are dead fat cells replaced by calcium, and this has a chalky white appearance. In fibrinoid necrosis, I want to hear you guys say that there are thick and pink vessel walls. And in gangrenous necrosis, we have two types, dry and wet. Okay, I'm going to give you guys how long? How long do you guys need? 2 minutes? 1 minute? Okay, I'm going to give you guys 1 minute. How the question will be in fibrinoid necrosis. The questions for fibrinoid necrosis is that they can tell you that there is presence of fibrinoid necrosis in the blood vessels, or they can give you a picture for fibrinoid necrosis, and if you see anything of this sort, okay, you can assume that there is fibrinoid necrosis going on. Another way that they can give you questions is by hypertensive urgency. For hypertensive urgency is characterized by excessive loss of the plasma proteins and accumulations and hardening. So that's another one. That's that. I'm going to give you guys 2 minutes. Hello. Oh, hi. How are you? Yes, I'm doing good. Thank you. Yes, he's doing comparatively well. His wound is it's actually getting better. There is very minimal leakage, and we're just changing the pack once a day, and everything else seems fine except the fact that his Larry tubes and HMEs have still not come, and we realized that Northwell, speech therapist that is PhD in, she would be responsible for sending those things. So we apologize for asking you the last time and to put you through all that trouble. So, well, she sent us 6 HMEs, so we are currently using those, but eventually we would be needing a bit more. Right. Yeah, right. 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 And I was also wondering, is it possible to make an appointment with a pulmonologist? I've been trying to do that, but is there any way that you can help me out? My father, he doesn't have any fixed pulmonologist as of yet. No, we don't have anyone in mind to as of yet, but I was wondering if there is one, if there's like anyone who can come to the house who can do the thing, or do we have to take him to the clinic? Okay, okay, all right. So, is it possible for you to make the appointment? I think. Okay, okay. So, I think that we're just gonna try to do it ourselves then. See what. Okay, so we're just gonna see that then. Yes, we are going to. Yeah. Yeah, we're going to. Oh, okay, perfect. Oh, okay. All right. Perfect. Thank you so much. All right. Bye-bye. Okay, is anyone ready? Is anyone ready for the necrosis questions? Okay, Dr. Tareen is ready. It's been a while, Dr. Tareen. Hope you're doing good. Let's hear the histologies of coagulative necrosis.
The histology for coagulative necrosis, there's preserved cellular, but the nuclei disappear.
So preservation of cellular architecture is there and nuclear disappearance is there. Perfect. That's for coagulative necrosis. What would you look for in a patient of liquefactive necrosis?
Neutrophils.
Neutrophils, very good. And cystic spaces is there. Next one is if you have a patient of caseous necrosis, what do you expect to see?
You see macrophages, and then it's like walled off.
It walls off, and in the middle, what do we have?
Fragmented cells.
No, in the
Wait,
middle.
in the middle, you have like
In the middle, we have the
you
caseous
have the actual organisms, right?
right. We have the caseous necrosis and the infecting organism. Very good. What do we see in fat necrosis?
You see saponification.
We see saponifications, and if we do some sort of a scan, what do we expect to see over there?
You see like
Chalky white appearance.
yeah.
Okay, so we see chalky white appearance. Fibrinoid necrosis. What do we see in the histology of fibrinoid necrosis?
The walls are thick and pink.
Walls are thick and pink. Gangrenous necrosis. How many types of gangrenes do we have?
There's two. The dry is ischemia without infection, and the wet is ischemia with infection.
Okay, perfect. So there is one without infection, that is dry. Another one is with infection, that's wet. Very good. Thank you so much, Dr. Tarian, for participating.
You're welcome.
And that's all for the necrosis. Okay, now, before we move forward to ischemia, do you guys want to take a break, or do we continue with ischemia? Ischemia, okay. Continue, okay. So let's get done with this page. This is not a very hard page to read. Then we would take the break afterwards, okay? Now, ischemia. Ischemia is inadequate blood supply to meet demand. Basically, the ischemia is basically, for example, if there is hypertrophy of the left ventricle has so much mass that it's not easy for the blood to reach all the parts of the left ventricle when it needs it. So when it needs it means demand. So what happens is there's loss of blood supply to certain regions of the left ventricle, and those regions can undergo ischemia. For example, this is the one that they're trying to show over here, that this is the portion where blood is not reaching because of the increased size of the left ventricle, most probably due to long-term hypertension. So that's that. So mechanisms will include decreased arterial perfusion, example, atherosclerosis, or decreased venous drainage, for example, testicular torsions or Budd-Chiari syndrome. Regions most vulnerable to hypoxia and ischemia and infarction. This is extremely high yield, guys, okay? Always remember that the regions that are extremely high yield to ischemia are the watershed region. What do we mean by the watershed region? What do we mean by the watershed region? Guys? meaning that there's the region of anastomosis, the region where their anastomosis happens. Okay, so for example, in brain, we have anterior cerebral, middle cerebral, posterior cerebral bounded arteries. Okay, that the regions of anastomosis, for example, those areas are extremely high yield. In the heart, we have the subendocardium. That is the layer that is just beneath the endocardium, subendocardium, because the blood supply of the heart comes from here to here, like this. Okay, right? So blood will reach these areas, blood will reach these areas, but it may not reach these areas. That is right below the endocardium. So subendocardial areas. Then in the kidneys, we have the straight segmented proximal tubule in the medulla and the thick ascending limb of the loop of Henle. Okay. In the liver, we have the central zone, zone 3. Okay. Do you guys remember zone 1, 2, 3 of the liver? Who remembers zone 1, 2, 3 of the liver? Yes? Okay. Okay. Which type of disease is more common in zone 3? Which type of disease is more common in zone 3? Most diseases. Okay, that's a very good answer. Which types of disease are alcohols and drugs? Alcohol, drugs, ischemic hepatitis, alcoholic hepatitis. Okay, so that is more common. Okay, colon. In the colon, we have splenic flexure. Splenic flexure, where it's also known as the Griffith point. It's not a very common term to use. And then rectosigmoid point is known as the Sudak point. This is a pretty common term. So the thing is, these are the two areas of anastomosis. Okay, these are the two areas of anastomosis. Okay, zone 2 is yellow fever. Very good. Zone 2 is yellow fever. Zone 3 is alcoholic hepatitis and ischemic hepatitis. And zone 1 is viral hepatitis. Very good. Okay, the liver. So that's that. Yes, viral or cocaine abuse. Okay. Now, before we move on, this is something which you guys have to realize and memorize. Okay, in the brain, ACA, MCA, PCA, heart, subendocardium, kidneys, straight segment, proximal tubule is more high yield, and then you have the medulla. Okay, then liver is central zone 3, and colon is the splenic flexure. Okay, I'm going to give you guys 1 minute. I'm going to give you guys 1 minute before I move on, because we are done doing homeworks. We are going to learn first aid right away in the lecture. Whoever is ready, please tell me you're ready. Whoever is ready to tell me the names. Okay. Okay, Dr. Hassam, let's hear you say the name, the areas. Okay, Dr. Hassam.
Yes, basically watershed areas. So in
Yeah.
brain, between the anastomosis of anterior cerebral artery, middle cerebral artery, posterior cerebral artery. In heart, subendocardial region mainly. In kidney, PCT,
Me?
proximal convoluted tubules, and thick ascending lobe of Henle are more
Okay.
predisposed.
Okay.
Then which organ? In colon, we have splenic flexure and rectosigmoidal anastomosis.
Very good. And in the liver?
In the liver, zone 3 mainly.
Very good. Zone 3. Zone 3 is the area of infarction. Is everyone clear about these areas? You guys will get tested on this left and right. Okay. Watershed areas receive blood supply from most distal branches of two arteries with limited collateral vascularity. These areas are susceptible to ischemia. Okay, they're just telling us something which we already know. Okay, so we are smarter than them. Neurons most vulnerable to hypoxic ischemic insults include, okay, this is high yield. But this is not something which we are reading right now. When we read neurology, do you remember I already mentioned this? The Purkinje cells of the cerebellum and the pyramidal cells of the hippocampus, the neocortex, right? So this is just a revision. Basically, in the brain, in the cerebellum, the Purkinje cells are more viable to get damaged. And in the cortex, you have the pyramidal cells of the hippocampus. If the pyramidal cells of the hippocampus are more damaged, can we assume that patients of ischemia are more at risk of having memory problems? Okay. Now, types of infarcts. How many types of infarct do we have? We have a red infarct, we have a pale infarct. Red infarct means that there is venous occlusion. Focus on this one. There's venous occlusion and tissues with multiple blood supplies with multiple, and this happens in tissues with multiple blood supply. Basically, you guys know very well that there's an artery, then there's a capillary, then there's a vein. Okay, right? Arteries, capillaries, veins. If you have venous occlusion, okay, then all this blood, will it not pile up in the artery? Yes or no? All the blood. So the blood that piles up in the artery, what is the color of the blood that piles up in the artery? Red. So the infarction is a red infarction. If you ever get confused about red infarct, is it an arterial occlusion or is it a venous occlusion? Can you please remember that venous occlusion will result in arterial blood accumulation, and that will be a red infarct. Okay, they will try to confuse you with red infarcts being arterial or venous occlusions. Okay, so that's that. Next one is pale infarct. Pale infarct occurs in solid organs with a single blood supply. Example, heart or kidneys. For example, over here, as you can see that there's a pale infarct, that over here, there's a solid organ or a compact organ with a single blood supply. Can we also assume that there's some amount of coagulative necrosis happening over here? Yes. Okay. So pale infarcts are basically single solid organs with an arterial occlusion. So there's arterial occlusion. As a result, if there is arterial occlusion, will any blood go to this region? No. If no blood goes, can we assume that that region will be pale? Yes, good, very good. Okay, and arterial occlusion. Next one. Next one is free radical injury. Okay, free radical injury. How many of us are accustomed to free radical injuries? Everyone should be accustomed to free radical injuries because this is the first time we're talking about free radical injury. No. Where do we talk about free radical injury? Okay, very good. NPDH. Okay, NADPH oxidase. Basically, we talked about free radical injury and chronic granulomatous diseases, right? Over there. NADPH oxidase, if it's not there, this can result in free radical injury, and that's that. Okay. And okay, so free radicals, okay, free radicals, they damage the cells via, okay, I want you guys to put your attention to these things: membrane lipid peroxidation, protein modification, DNA breakage, and these are the three things: membrane lipid peroxidation, protein modification, and DNA breakage. NBME will choose these three phrases to direct you to free radical injury. Okay, initiated via radiation exposure is an example, metabolism of drugs is an example, and we have all other types of different examples, things which you do not have to put your attention in, so you can skip this out because anything and everything can cause free radical injury, more or less. Free radicals can be eliminated by scavenging enzymes. Examples are catalase, superoxide dismutase, and glutathione peroxidase. Okay, do you guys remember that process of GSS converting to GSSR, right? GSSG, right? So that's the one. Then there is spontaneous decay, antioxidants, and certain metal carrier proteins can also cause free radical injury. But these are the three things that prevents free radical injury: catalase, superoxide dismutase, and glutathione peroxidase. Examples of free radical injury is oxygen toxicity. For example, rib. Okay, in rib, what was rib? What was rib? Who remembers rib? RIB. Well, who remembers rib? Rib for R for retinopathy of prematurity, I for intraparenchymal hemorrhage, right? Or intraventricular hemorrhage. B for bronchopulmonary dysplasia. These are signs symptoms of oxygen toxicity in a young patient. So retinopathy of prematurity is there, characterized by abnormal vascularization, bronchopulmonary dysplasia, reperfusion injury after thrombolytic therapy. These are the examples. You do not have to focus on this, so don't worry about this. Drug toxicity, acetaminophen overdose, carbon tetrachloride, and they can also induce free radicals that can cause free radical damage. And decreased apolipoprotein synthesis can also cause free radical damage. Metal storage disease, hemochromatosis, and Wilson's disease, these are also examples of free radical damage, characterized by a lot more other free radical damages that are there. Basically, any one of these things will be there in your question stem. That is, if they want to make a question about free radicals, then they will choose any one of these examples. They can have the question made about hemochromatosis. They can have the question made about Wilson's disease. They can have the question made about oxygen toxicity or anything. But the answers are basically either they can ask you, what do you expect to see in the histology? In the histology, you can see membrane lipid peroxidation. Whenever you identify the questions about free radical injury, this is what you're looking for. Okay, membrane lipid peroxidation, protein modification, and DNA breakage. Okay, I will give you guys 1 minute to read infarct and free radical injury. I want you guys to read these things, these three things, and keep in mind that there's membrane lipid peroxidation. Basically, if I have to talk about membrane lipid peroxidation, that is the lipid membrane is the phospholipid bilayer. If it gets oxidized, then there is damage to the membranes that can result in membrane breakage and loss of cytoplasmic contents. Protein modifications can happen, resulting in loss of protein synthesis. And obviously, DNA breakage will interrupt the whole process of protein synthesis by preventing DNA transcription, translations, and all those things. So that's that. And another three things which I want you to remember is anti-free radical damage. That is catalase, superoxide dismutase, and glutathione peroxidase. These are the three things which I want you to remember. I'll give you guys exactly 1 minute, and in that 1 minute, I'll ask you guys whether you guys can remember free radical damage or not. Okay. (12:13) as of right now. I'll ask you at exactly 12:15. Please read infarction. Please read free radical damage. Whoever is ready, please tell me you're ready. Dr. Time is ready. Okay, perfect. Let's wait for a little bit more time. It's 12:14. I'll ask you at exactly 12:15. Who else is ready? It's 12:15. Dr. Grabasi is ready. Who else? Okay. So let's ask the question to Dr. Grabasi. Dr. Grabasi, how many types of infarcts do we have?
Two types.
What are the types of infarcts?
Red and pale infarcts, and red infarcts occurs like due to venular occlusion and like multiple blood supply, for example, like the liver or the lung, the testis, or the intestine.
Okay.
And the pale infarct is like to like a single arterial supply, for example, in the kidney.
Okay. Good.
Yeah.
Those are reds and those are pale infarcts. You answered all of them even before I could ask you the question. So good job with that. Next one is free radical injury. What do we expect to see in free radical injury on histology?
Like lipid membrane peroxidation.
That's number one, membrane lipid peroxidation. Number two is?
Protein modifications.
Number two is protein modification. Very good. Number three is?
And DNA breakage.
DNA breakage. Very good. And what are the three enzymes that prevents free radical damage?
Catalyzed superoxide dismutase and glutathione peroxidase.
Glutathione peroxidase. Very good. So thank you so much. So these are the things which
You're
I want
welcome.
you to remember from over here. Hope every one of you identified these things, okay, so that it's easy for you to do a quick revision and recapitulation in the future when you are reading it again during a dedicated period. Now we're going to move on to types of calcification. Okay, are we ready to take the break right now, or are we going to take the break after we come back? Which one? Break. Continue. Continue. And then? Okay. Okay, how about we do take a break since most of you guys are saying break. Okay, so let's take a break for not longer than 10 to 15 minutes. Let's come back at 12:30-ish. Okay. Let's come back at 12:30-ish because it's already been more than 2 hours we've been doing the lectures. Your neuronal firing should have decreased by now. Okay. We need to give our synapses enough time to fill with dopamines and serotonins, right? And all the excitatory neurotransmitters and glutamate. Okay, so you have to give your synapses some time. 10 to 15 minutes is more than enough. Get your synapses ready because after we come back, we will begin with calcifications and amyloidosis, and we will be done with pathology, the first segment. Then we will try to move on to inflammation. Yes, what is your question? Will we be finished by this section tomorrow? Do you mean pathology? Yes, okay. I want to, personally, I do, because I am hoping that next week will be the last week of the lecture before we begin our new batch. So I'm gonna try to do my best to finish all of these things. Okay, I'm gonna try to do my best to finish it by tomorrow. If not tomorrow, then hopefully by next week, by hook or crook, I'm gonna try to finish as much as possible. Okay, are we clear? I don't understand what you wrote to me. Where did I write? Where? Oh, okay. See, that's why we need breaks, because we are not very, okay, that's why we need breaks. I don't understand what you wrote to me. Also, for a student that messaged me. Okay, good. So we shall take the break and we will come back in. It's 12:19, so we'll come back at 12:30, 12:31. Okay, see you guys in 10 minutes. Okay, is anyone back from their break? It's 12:31. Yes? Okay, perfect. Okay, so let's begin with our discussion of first aid. That is, we are going to discuss about calcification. So let's talk about the types of calcification. Okay, now calcification. Calcification always appear deeply basophilic, as you can see over here. If you do histology on a hematoxylin and mucin stain, then it will appear as deeply basophilic. If I talk about a tissue with PAS positive deposition, which type of deposition am I talking about? PAS positive deposition. Let's see who can give the first answer. Very good. Glycogen. Very good. Okay, so every type of deposition has a different type of indication. So calcification and calcium deposition is deeply basophilic. For example, glycogen deposition is positive for periodic acid shear stain. So that's that. Now, dystrophic calcification and metastatic calcification. Okay, dystrophic calcification always happens in diseased tissues. Extent is it's always localized. For example, with old age, with the wear and tear of aortic valves, okay, there's deposition of calcium. And whenever there's deposition of calcium in aortic valves, this can give rise to senile aortic stenosis due to calcium deposition. This is known as calcific aortic stenosis. Okay, in the associated conditions, you have tuberculosis, where there's lungs and the pericardium that are affected, and other granulomatous infections, liquefactive necrosis of chronic abscesses, fat necrosis, infarcts, thrombi, schistosomiasis, and blah, blah, blah. And all of these things, they can get calcified, not high-yield, associated conditions, okay? Tuberculosis and other granulomatous, basically what they're trying to say is that in any type of necrosed or diseased tissue, you can have deposition of calcium, as these examples are given over here. Etiology is secondary to injury or necrosis. Basically, after necrosis, the cells, the calciums, they go and they deposit in those regions. Okay. Next one is serum calcium level over here is normal. That is, the calcium level is not changed. Okay. The reason why we would like to believe that there's calcium deposition is if I tell you that isn't there a possibility that in these tissues in dystrophic natures, is there a more acidic medium or alkaline medium for deposition of calcium? Everyone? Calcium deposits in acidic medium or an alkaline medium? If calcium deposited in acidic medium, then why do we not have stomach calcified stomach, right? So calcium deposition never happens in acidic medium. Calcium deposition always happens in alkaline medium. So if there is a dystrophia, then we can assume that the surrounding medium is an alkaline medium, and then there's deposition of calcium. So there's secondary to injury necrosis. That's that. Metastatic calcification, it happens in normal tissues. It's a widespread, it's not localized, and it's predominantly in interstitial tissues of the kidney, lungs, and gastric mucosa, and the increased pH favors calcium deposition. It says so over here. Okay. Okay, these tissues, they lose the acid quickly, and as a result, there's metastatic calcium deposition. So which tissues are they? Kidney, lungs, and stomach. Lungs, kidney, and stomach. Okay, lungs, kidney, stomach. They lose acid more quickly. So nephrocalcinosis of collecting ducts may lead to nephrogenic diabetes insipidus and renal failure. This is an example of metastatic calcification. Etiology is secondary to hypercalcemia. So when you compare these two, you can see that the calcium level over here is normal. The calcium level over here is abnormal. Okay, and it's due to high calcium or high vitamin D. Because with vitamin D, if it's high, then there's increased gut absorption of calcium. So that's how you have hypercalcemia due to hypervitaminosis, or high calcium phosphate product levels, example, CKD with secondary hyperparathyroidism, long-term dialysis, calcified excess, and multiple myeloma. These are all the causes of high calcium depositions. Okay. Now, if can you expect a question with tables from these two calcifications, dystrophic calcifications and metastatic calcification? What sorts of tables can you see? Okay, basically over here, they can ask you a question whether the extent is localized or rare, or it's widespread. Okay, they can ask you a question whether the calcium is normal or abnormal. Okay, and they can ask you questions regarding the etiology. Okay, without even trying to read this based on my discussion, if I can, is anyone confident to answer some questions about dystrophic calcification and metastatic calcification without reading this? Because these are very easy, direct questions. Okay, so who is confident? Dr. Wassam is confident. Who else is confident? Dr. Tharin is confident. Okay, let's ask you two. First of all, let's start with Dr. Hussam. Dr. Hussam, dystrophic calcification. In which types of tissue can you expect dystrophic calcification?
In necrotized tissue.
And Dr. Tharin, which type of tissues can you expect to have metastatic calcification?
In normal tissue.
Okay, dystrophic calcification, Dr. Hussam, is it localized or is it widespread?
Dystrophic localized.
And so metastatic is widespread. Next one is Dr. Tharin, would you tell me the causes of metastatic calcification?
Hello, can you hear me?
Yes. Well, what are the causes of metastatic calcification?
Nephrocalcinosis.
No, that's not a cause. That's an example. Causes.
Oh, like what's causing it to be
Yes.
deposited?
Yes.
The pH.
Okay. High pH and
Like
basically all the
tissue
conditions.
being disrupted.
No, destructive tissues will have dystrophic calcification. Metastatic calcification, the etiology is high calcium. All the conditions leading to hypercalcemia is responsible for metastatic calcification.
Okay.
Okay. Dr. Hussam, what happens with the calcium level in dystrophic calcification?
Normal.
Dr. Tharin, what happens with the calcium level in metastatic calcification?
It's high.
Yes, it's high, it's abnormal. Can you name 3 organs where metastatic calcification is more prone?
The kidney.
Kidney.
How
about the stomach?
Yeah, the stomach.
Okay,
And
the stomach, kidney, and
what about the lungs?
Yes, lungs.
Right?
So stomach, lungs, kidney, and stomach. Very good. Okay. So these are the 3 places. So thank you, Dr. Hussam and Dr. Tharin, for your contributions to participation. Next one is lipofusion. Lipofusion is let's talk about lipofusion because you will get a lot of questions about this one. Okay, because the thing is, you will try to confuse lipofusion with heart failure cells. Okay, heart failure cells and lipofusion, they look the same. Okay, who knows heart failure cells? Are we well aware of heart failure cells? What is the another name for heart failure cells? What is the another name of heart failure cells? Hemosiderin-laden macrophages. Very good. This is hemosiderin-laden macrophages. So basically, heart failure cells are when the left ventricle fails to contract and blood piles up in the capillaries of the pulmonary vasculature, and those capillaries filled with blood leads to leakage of blood and phagocytosis of the blood by the macrophage, resulting in heart failure cells or hemosiderin-laden macrophages. Students have the tendency of confusing heart failure cells with lipofusion. The way that you can avoid the mistake is by, because the picture all more or less looks the same, the way that you can avoid the mistake is by looking at the question stem. If the question stem is talking about a person with a normal aging with no associated pathology, then it's lipofusion. That is a yellow-brown wear and tear pigment. It is associated with normal aging. It is composed of polymers of lipids and phospholipids complexed with protein. So lipids and phospholipids complexed with protein. It may be derived through lipid peroxidation of polyunsaturated lipids of subcellular membranes. Basically what happens is that the lipids over here, they can undergo the process of oxidation, and after oxidation they can result in the formation of this red brownish like depositions known as lipofuscin. So whenever you see lipofuscin in a tissue, you can imagine that this is the tissue of a very old person, okay, because this is a senile change. Autopsy of an elderly person will reveal lipofuscin in heart, colon, liver, kidney, eye, and other organs. So that's it. So once again, lipofuscin, it's not a harmful inclusion. It will not do anything, but lipofuscin is basically an indicator of normal aging. Are we clear about lipofuscin? Yes or no? Okay. Now, amyloidosis. Okay, let's begin our discussion with amyloidosis. So if old age, we suspect lipofuscin, yes. Do you guys have any questions till now? Any questions?
So basically, how you would tell the difference is based on the stem of the question.
Right, by
Okay.
looking at the question stem, okay, more or less. Okay. Normal aging only? Yes, normal aging only. Okay. Any other question? Does anyone have any other question? Can I move on to amyloidosis? Okay. Now, amyloidosis, okay? Now, amyloidosis is abnormal aggregation of proteins or fragments into beta-pleated linear sheets, okay? This results into insoluble fibrils, cellular damage, and apoptosis. Okay, so what happens is amyloids are a type of protein, okay? Amyloids are a type of protein where the protein is transformed into a beta-pleated sheet. Basically, if it's an alpha pleated or if it's an alpha amyloid, this can easily get broken down by proteases, and they will not result in accumulation of the protein. But whenever you have accumulation of any substance beyond the scope of a tolerance of an organ, can it cause damage to the organ or hamper the function of the organ? Yes or no? Yes. Can we say the same thing about metastasis or tumor metastasis, right? That whenever there's excessive accumulation of excessive accumulation of it. So the thing is, whenever there is a protein that is converted into a beta-pleated sheet, we do not have any protease in the body that can break down this beta-pleated sheets. As a result, you have amyloids. Amyloids are visualized by Congo red stain. In Congo red stain, you can have red-orange on non-polarized light and apple-green birefringence on polarized light. So if you can see over here, this is apple green, right? This is apple green, and this is the Congo red. This is the Congo red stain over here. Okay, in the Congo red stain, you can see that there is amyloids like this one, and in the non-polarized light, you can see that there's an apple green birefringence. So they can use any one of these ones over here, or hematoxylin and leucine stain is also there. So in hematoxylin and the leucine stain, you can also see a little bit of about a little bit of amyloid. But amyloidosis questions are always they always contain Congo red stain in their associations. So whenever we see Congo red stain, the first thing we assume is amyloidosis in the question stem, basically. Okay. Tubular basement membranes are enlarged on light microscopy if we do a hematoxylin and leucine stain, if there is nephrogenic amyloidosis or nephroamyloidosis. Okay. Now, How many types of amyloidosis do we have? Systemic. What do we mean by the word systemic? Systemic means that there is amyloidosis in different cells or tissues of the body, different organs or tissues of the body. Localized basically means there's one particular organ where there is accumulation of the amyloids. And hereditary are the conditions where, due to certain conditions, there is an increased predisposition of result of having amyloids. So let's talk about systemic amyloidosis. That is that there are three types: primary, secondary, and dialysis-related amyloidosis. Basically, systemic amyloidosis, once again, is the deposition of amyloids in different tissues of the body. Okay, so number one is primary amyloidosis. The fibril protein that is accumulated is AL from immunoglobulin light chain. It's seen in plasma cell disorders. For example, multiple myeloma is a type of primary amyloidosis. So primary amyloidosis, PL, for plasma cell disorders and L chain deposition. Secondary amyloidosis is serum amyloid A is the one that is accumulated over here, and it is seen in rheumatoid arthritis, IBD, familial, this is not high yield, familial Meridian fever. Okay? If you can only remember rheumatoid arthritis and IBD, that's enough. And dialysis related amyloidosis, basically what happens is that during dialysis, during dialysis, when the blood is being cleaned off, there could be presence of the proteins of the body, they can take a change into a beta microglobulin-like structure, which is a bit difficult for the body's proteases to break down. As a result, you have depositions of these type of proteins, which are amyloids, that is beta 2 microglobulin form of amyloids that gets deposited in different tissues of the body and causes damage. So in dialysis, you can have amyloidosis that is known as dialysis-related amyloidosis due to the deposition of beta 2 microglobulin. It's seen in end-stage renal disease or long-term dialysis. The manifestations are basically, these are the manifestations of what would happen if these amyloids deposit in different tissues of the body. Let's talk about the heart. If there is deposition of amyloids in the heart, it will cause restrictive cardiomyopathy. If there's depositions in the GI tract, this can cause hepatomegaly. Macroglossia is basically increased shape and size of the tongue. In renal, if there's amyloidosis, it's high yield for causing nephrotic syndrome because it causes membrane damage and lack of fluid, loss of plasma proteins. In hematological, it can cause splenomegaly. Neurological, it can cause neuropathy. Musculoskeletal, it can cause carpal tunnel syndrome because if there's deposition in between the layers of in between the tendons through which the median nerve goes through, there could be compression of the median nerve, and this can give rise to carpal tunnel syndrome. Okay, so before I move on to localized, I'm going to give you guys 2 minutes to learn primary, secondary, and dialysis-related amyloidosis. Are these examples high yield? The thing is, these examples, they can come in the presence of the fact that whenever they talk about amyloidosis and for example, they can associate a question of restrictive cardiomyopathy with these sorts of presentations. So you have to know that amyloids are responsible for causing restrictive cardiomyopathy. And they can also attach questions of nephrotic syndrome with the renal depositions of amyloid. So there will not be any direct questions, but there will be associated questions. So yes, it's high yield. About the stains, maybe sometimes we have to identify accordingly. Yes, we do have to identify accordingly. So this distinctive thing is fibril protein. Fibril protein is basically amyloids are of all the different types of protein architectures, fibrillar type is the type of amyloid protein that gets accumulated. So all types of amyloids are of fibrillar types. Okay, any other question? Does anyone have any other question? I don't understand the concept behind dialysis-related amyloidosis. Whenever a patient undergoes dialysis, there's a possibility that certain proteins in the body, they change and take up the form of they take up the form of a beta-2 microglobulin. So basically, what happens is there's accumulation of amyloid fibrils, which are beta-2 in the bones, and the viscera of the patient who is undergoing dialysis. So basically, it's a very uncommon sort of a presentation. Not a lot of patients will have dialysis-related amyloidosis, but if there is a patient who is undergoing dialysis and then all of a sudden there is onset of symptoms of multiple organ failure or multiple organ dysfunction, then we can assume that due to the process of dialysis, there is a change in the structural protein of the body, maybe due to stress. It may be due to, because do you understand that in the process of dialysis, there's a lot of different types of stress that the body goes under. So maybe due to stress, the exact mechanism of the change of the structure of the protein is not known. But what happens is in long-term dialysis, there's change of the structure of protein from a normal structure to a fibrillar beta-2 microglobulin-like structure, which causes the disease. That is dialysis-related amyloidosis. Is anyone ready? Okay, is anyone else ready? Can we have some participation from other students? Okay. Dr. Naud, okay. Dr. Naud, how many types of systemic amyloidosis do we have? 3 types, okay. Primary amyloidosis, what is the form of fibrillar protein that gets deposited? AL. What is the type of secondary amyloidosis protein that gets deposited? Okay, what is the type of dilated dialysis-related amyloidosis that gets deposited? Okay, what are some of the cases of secondary amyloidosis? Okay, good. Okay, now, can we move on to the next one, localized amyloidosis? Okay, let's move on to the next one. Okay, before we do that, if we have depositions of amyloids in the heart, what would happen? Everyone? Restrictive cardiomyopathy. If you have depositions in the renal system, what would happen? Okay, if we have depositions in the brain, what would happen? Brain problems. A very good answer. Okay, how about if we have depositions in the musculoskeletal, what would happen? Carpal tunnel, very good. So that's that. Now let's talk about localized amyloidosis. First one is Alzheimer's disease. Okay, we all know that in Alzheimer's disease, we have beta amyloid protein that gets accumulated, right? Beta amyloid protein gets accumulated, which is cleaved from amyloid precursor protein. So the beta amyloid protein gets accumulated. In type 2 diabetes, we have islet amyloid polypeptide. Unfortunately, there's not much you have to understand. All you have to do is learn and memorize. So that's that. But this should not be very hard, because is this the first time we're talking about beta amyloid protein accumulation in Alzheimer's disease? Yes or no? No, right? Exactly. Now, next one. Next one is medullary thyroid cancer. We have deposition of calcitonin, which is a form of amyloid. Atrial natriuretic peptide is also a type of amyloid which can cause isolated atrial amyloidosis. Now, let me ask you this. Let me see if we can get a solid answer. If you have isolated atrial amyloidosis, will there be more functioning of the left atrium or no functioning of the left atrium? Okay, there will be very decreased functioning of the left atrium. So as a result, can you have pulmonary hypertension? Okay. Okay, now let's talk about systemic senile amyloidosis. Systemic senile amyloidosis. This is normally seen with transthyretin. Transthyretin is a type of amyloid that gets deposited over there. Okay, I'm going to give you guys 1 minute again. Okay, all you guys have to do is just learn the names of the amyloid which gets deposited. Nothing else. Okay, nothing else. So 1 minute on the clock, it's 12:57. I'll ask you at 12:59. Okay. Dr. Hassan is ready. Okay. Now, Dr. Hassan, what do we see in Alzheimer's disease? Which amyloid protein is accumulated? Beta amyloid. Which one do we see in type 2 diabetes? Islet amyloid precursor protein. Which one do we see in medullary thyroid carcinoma, Dr. Hassan? Calcitonin. Which one do we see in isolated atrial amyloidosis? ANP. Which one do we see in systemic senile amyloidosis? Transthyretin. Very good. Okay. Were you looking at the book when you gave the answers? Okay. Perfect. Thank you. So I trust you and thank you so much for participating. So that's that. Is everyone clear? Yes? Okay, perfect. Now, let's go to the last one, that is hereditary. So there are two types of hereditary conditions when you can have depositions of amyloid, and comparatively, these are not high yield at all. So I don't want to put a lot of emphasis on this. You have number one, that is familial amyloid cardiomyopathy, where there is depositions of transthyretin. So in all cases of familial forms, you have the easiest thing is the type of amyloid that will get deposited is a transthyretin form, both in familial amyloid cardiomyopathy and familial amyloid polyneuropathy. So that's that. And of course, if you have deposition of these types of amyloid proteins, for example, the one that will happen in the heart, if there's deposition of amyloid protein, there should be restrictive cardiomyopathy. If there's deposition of amyloids in the brain, this can cause polyneuropathy. So that's that. Okay, not very high yield, because I'm going to tell you why. Because they will have the mention of the fact that there is familial amyloid cardiomyopathy in the question. They will not ask you to diagnose this. Okay, they will not ask you to diagnose this. Are we clear about this? Yes or no? Okay. So if you have to see a amyloid on histology, Dr. Adhanom, can you hear my question? Can you hear me out? Dr. Adhanom? Can you unmute yourself? If you want to see amyloid on a tissue, which types of staining would you do?
Yeah. Congo red stain.
Very good. And over there, what would you see in polarized light?
By refrigerants, apple green.
Very good. What would you see in non-polarized light?
Non-polarized light.
Yes. What would you see?
I mean,
You will just
HND
see
stain.
deposition. You will just see depositions of amyloid like this one in non-polarized light.
Okay.
Okay. Are we clear about this?
Yes, yeah.
Okay, perfect. My apologies for asking you all of a sudden. Just want to let everyone know that I'm going to ask you these questions out of nowhere. So everyone should be ready. Okay. Now, the thing is, yes, Dr. Hussam, what is your question? Yes, everyone has a question. Dr. Hussam, what is your question? Birefringe, meaning that, okay, that's a good question. What is birefringe? Apple green birefringe, meaning that there's an area of color, then there's an area of discolor, then an area of color, then an area of discolor, something like this. This is called birefringe. Okay. See, area of color, area of discolor, area of color. That's for Dr. Hussam. Yes, Dr. Tarun, what is your question?
For the localized amyloidosis, besides systemic amyloidosis, at the side there's like a note saying cardiac dysfunction is, I guess, more slower onset than the AL. Can you just explain that a little bit?
Okay.
Is that even important? I don't know.
Okay. So this is age-related amyloidosis, meaning that with age, what happens is there is decrease in the protease activity. So normal types of transthyretin, which are easily degraded, they do not get degraded, so they start accumulating, and predominantly they start accumulating in the cardiac ventricles. And the reason why that is because with age, not a lot of the tissues and organs of the body are functioning as highly as the heart, because the heart is still responsible for pumping the blood. So with age, what happens is that the regeneration process gets slower, and also the proteolytic enzymes, they get decreased in number. So you have accumulations of these things. With damage, you have accumulations. So that's why you have damage in the cardiac. That's why you have accumulation in the cardiac ventricles. And as a result, this can cause cardiac dysfunction, which is more insidious than in AL amyloidosis, that is in primary amyloidosis. This can happen very gradually, but this one is more insidious, meaning that this can happen all of a sudden. And no, this is not high yield. That's what it is. Okay, this is not high yield. Okay. Now that inflammation. Okay. Do we want to start with inflammation, or do we want to do some questions? Which one do you guys want to do? Question. Okay. So if you want to do questions, can I give you guys a small homework for today? Can I or can I not? Yes or no? A small homework. Can we do inflammation because it's similar to immunology? Okay. Be fast. Okay, we'll try to do it, but let me just give you guys a small homework for today. Okay. Now, I want to give you guys a small homework over here. I want you guys to read 1, 2, 3, 4, 5, 6. Only this 6. There's not really much to understand from over here. All you have to do is learn the name, which type of receptor it is, and which type of carcinoma will it cause, oncogenes. So that the next time when we do this, by the time we come here, we will be done. Okay, so homework. It will not take you more than half an hour. Not even half an hour. It should take you around 15 minutes. Okay, are we clear? Okay. So tomorrow morning, we'll start with this one. Okay, now, so Inflammation. Let's start with a little bit of inflammation, and then we can do a little bit of question, and then we can end this in a little bit. Okay, now so let's start with the first one, that is inflammation. Not really much to understand because inflammation is very easy. Inflammation is the response to eliminate initial cause of cell injury, to remove necrotic cells resulting from the original insult, and to initiate tissue repair. It's divided into acute and chronic. The inflammatory response itself can be harmful to the host if the reaction is excessive. That is, this line over here, that is, the inflammatory response itself can be harmful to the host if the act of inflammation is excessive, meaning that in septic shock, right? In septic shock that is due to interleukin 1, 6, TNF, we have septic shock, right? And due to endotoxins, over there, the amount of inflammation is so high because there are so many cytokines that are released all of a sudden. This can cause vasodilatation, life-threatening vasodilatation, loss of vascular tones, and this can cause shock and death. So this means that inflammation has to be there, but it has to be in a normal amount. Or if there's a prolonged inflammation, that, for example, in chronic inflammation such as tuberculosis, this can cause fibrosis and cellular dysfunction. Another one is inappropriate, that is in autoimmune diseases such as SLE. Now, what are the signs of inflammation? We have rubor, tumor, dollar, functional Lisa. Rubor, tumor, dollar, functional Lisa. Another one is Calor. I was looking for this Calor, rubor, Calor. So 5 signs of inflammation. Basically, rubor and Calor meaning redness and warmth. Why is there redness and warmth? It's very easy. First of all, when you have inflammation, you have vasodilatation due to the release of vasodilatory substances. The vasodilatory substances are histamine, prostaglandins, and bradykinin. Okay, this is a question, a very easy question, that which mediators are responsible for causing redness? Histamine, prostaglandins, and bradykinin. Swelling. Swelling is basically you have decreased adhesion in between the adjacent cells. So there is increased space between the adjacent cells, resulting in leakage of vascular fluids into the interstitial space. This results in swelling. The mediators responsible for swelling are leukotrienes, histamine, and serotonin. These are responsible for the swelling. Dolor is pain, meaning that there's sensitization of the sensory nerve endings. Do you guys remember the pain receptors as sensory nerve endings? Then we studied Merkel's disc and Meissner's corpuscles, Ruffini's corpuscles. Do you guys remember that? Messi had hair and this and that, right? From over there, do you guys remember that pain receptors are free nerve endings? Alpha receptors were there, and then then which other receptor was there? Delta receptor, very good. Okay, so over here, dolor is sensitization of the free nerve endings. Now, this is high yield, the mediators, that is prostaglandin, prostaglandin E2, histamine, and bradykinin. These 3 are responsible for causing pain. This is a question from NBME. Okay. This is a question from NBME too. Okay. Functional Lisa is loss of function, basically. So if you have so many things, you will have loss of function. These are the signs of inflammation. Basically, the next time when you have, for example, cut yourself, okay, you can see that the area that is cut will be red, swollen, indurated. They will have pain. And if there's a lot of pain, you cannot move that part. So there will be loss of function. So these are all the signs of inflammation. Systemic manifestations are, what are the systemic signs that happens due to inflammation? That is fever. Fever happens due to the release of interleukin 1, TNF. And which other interleukin can also cause fever? Very good. Interleukin 1, TNF, interleukin 6 can also cause fever. And it increases, what happens is that the pyrogens, it induces the macrophage to induce release of interleukin 1, TNF, and, excuse me, my apologies, increase the cyclooxygenase activity in the perivascular cells of the hypothalamus. As a result, you have increased temperature set point. So can we assume that fever is a protective mechanism of the body against infection? Yes or no? Because the bacteria will not thrive in high temperature. So what happens is your body's immune system, it increases the temperature set point of the hypothalamus, especially in the perivascular layer of the hypothalamus. And that's why you have fever, and this prevents bacterial inflammation, and it also acts as a sign of infection. Is fever a protective mechanism of the body or not? Okay. What would happen if we did not have fever? Would we be able to understand that we have an infection and have antibiotics? No. Okay, right? Exactly. So it's a very good indicator. Next one is leukocytosis. Leukocytosis is elevations of white blood cell count. Of course, if you have an infection, then there will be release of chemotactic factors and opsonizations and all of those things. And there will be increase in white blood cell count. Every white blood cell in the body will rise up and they will realize that there's a war going on. So they have to act up. So there will be leukocytosis. Type of cell that is predominantly elevated depends on the inciting agent. Of course, if it's a bacterial, it's neutrophil. If it's a viral, it's lymphocyte, right? Okay, next one. Next one is acute phase reactants. Then acute phase reactants will also be high. For example, the acute phase reactants are over here, positives and negatives. Guys, this is extremely high yield. This is high yield. Which ones will increase? Which ones will decrease? Ferritin, fibrinogen, you guys will get tested on this. They will paint out a scenario of an infection and they will tell you which one of the following things or which one of the following options, answer options, will be high. Over there, if you have any of the fish, that is ferritin, fibrinogen, serum amyloid A, hepcidin, or CRP, all of them should be high. Okay, more fish in the sea. Ferritin is an acute phase reactant. It binds and sequesters iron to inhibit microbial iron scavenging. Okay. Now, which acute phase reactant do we usually measure on a clinical setting or in a hospital setting to see if we have a CRP? Very good. CRP. So CRP is was more regularly tested to see if there is a bacterial infection or not. Okay, so ferritin will bind and sequesters iron. Why do they do that? Ferritin will take away the iron because the organisms, they have a tendency of using the iron to power themselves up. So they take away the iron. First of all, you take away the iron. Next one is fibrinogen is a coagulation factor which promotes endothelial repair. So fibrinogen tries to wall off any infection. It's a coagulation factor and it also promotes endothelial repair. Increased amount of fibrinogen is associated with increased ESR. Okay, increased ESR. You will realize when you get an infection or inflammation, the ESR is high. Serum amyloid A will prolong elevation, can lead to amyloidosis. Hepcidin will decrease iron absorption and decrease iron release. So do microorganisms, do they love iron or do they not love iron? They love iron. So should we give them more iron or less iron? Less iron. We should give them less iron by, first of all, increasing ferritin. Ferritin will take away the iron, and hepcidin will also increase. That will decrease the iron. That will decrease the iron absorption, basically. Okay, are we clear about this? Yes or no? Yes, okay. Next one is CRP. CRP is an opsonin. Opsonins will fix the component and cause phagocytosis. So opsonins, they are for complement fixation. What happens after complement fixation? After complements get fixated, of course, we have the activation of the membrane attack complex and formations of pores and destructions of the cells. So that's that. And that will also act as phagocytosis. Okay, it will cause opsonizations. It will also cause phagocytosis. That's that. It's measured clinically as a non-specific sign of inflammation. Things that will go down. Albumin will go down because we want to save the protein for later because these proteins can be broken down to provide amino acid that can be used for cellular regeneration. And transferrin will also go down because transferrin can increase the iron level. So they are internalized by macrophage to sequester iron. Okay, so transferrin have irons in them, so they are sequestered by macrophage. As a result, they are down. Okay, now can we ask can we ask Dr. Evelyn, would you be kind enough to name the acute phase reactants that are increased in infection or inflammation? Hey, Dr. Evelyn, can you hear my voice? Would you be kind enough to name the acute phase reactant, only the IL-1? IL-1 is not the acute phase reactant. It's a cytokine. The acute phase reactant that we just said. More fish in the sea. Fish. F4. More fish in the sea. F4. Okay, can anyone else name the acute phase reactants that are increased in infections or inflammation? Ferritin, fibrinogen, and then which one else? Serum, okay, and hepcidin. Good, very good. Which two are decreased? Albumin and transferrin. Very good. Okay, so that is that. Okay, now. Are we clear about this? Yes or no? Inflammation. So can we start the next ones from tomorrow and see how much we can finish tomorrow? Okay. For today, can we do some microbiology question to see where we stand in terms of microbiology? Okay, so I'm going to give you guys 5 minutes while you guys get freshen up, and I will upload the questions for microbiology. Okay, so can you guys hear my voice? Okay, so I uploaded the questions in the Facebook discussion group. Do we have anyone who has not joined the Facebook discussion group as of yet? Okay, so please join the discussion group where I can accept your invitation, and over there you can see the questions. If not, you can also see it from my screen. Okay, so you guys are more than welcome to go to the discussion group and download the questions, and we can discuss them right now, or you guys can view it from my screen. Okay. Now I'm going to share the screen over here. Okay. No. So can you guys see my screen? Can you guys see my screen over here? Okay. So let's begin with the first question. Let's begin with the first question. An outbreak of waterborne gastroenteritis with high mortality is reported in some Asian countries. It's caused by gram-negative, oxidase-positive bacilli that grows in alkaline media. Which of the following patients would require the smallest infectious dose? Now, whoever is ready, please tell me you're ready. Who else is ready? Okay. If everyone is ready, what is the correct answer? A. People who have been on chronic omeprazole therapy have low stomach acid content. And which organism are they talking about? H. pylori is not the correct answer. They're talking about Vibrio cholerae. Grows in alkaline media. Do you guys remember 42 degrees Celsius camping for Campylobacter jejuni, alkaline media for Vibrio cholerae, and urease positive for Helicobacter pylori? You guys remember that? Okay. Now, which one? Now the next one is a 60-year-old woman comes to the physician with 10 days of diffuse itching. A rash began on her hands, quickly spread on her wrist, arms, and axilla. She has difficulty sleeping. She works in a daycare center and takes care of children 2 to 5. She has no other medical condition. Vital signs are normal. BMI is 24. Skin shows this types of papules with excoriations, something to focus on. Vesicles, pustules in between, and also something to focus on here, that is webs, palms, and wrist. So if I were you, I would underline these things. Okay. Now, she has similar lesions in her axillary skin fold. Now, which of the following is the most likely finding on skin sampling? If anyone is ready, please tell me you're ready. Everyone else, are you guys ready? If I tell you that E is not the right answer, do you guys need more time? E is right? Yes, E is right. Okay, E is right. So that's the correct answer. The answer is never, not E. Very good. That's how confident I want you guys to be. Even if I tell you it's not scabies, you know I'm wrong. That is addition. Can you guys hear my voice? No. Okay. Yes, but okay. So the one we discussed on skin folds and immunity, but you have to realize but that they mention excoriations. You don't have excoriations on pediculus, on thyris pubis. Okay, you do not have excoriations. You know what excoriation means? No, no, I understand your answer is E, and I also understand the confusion. But do you understand the meaning of the word excoriations? Well, what do we mean by excoriation? Excuse me. My apologies. What do we mean by the word excoriation? Everyone is busy reading the question. Skin picking. Well, excoriation is basically the, right. Well, excoriation is scratching on the skin, but at the same time, there's also some sort of a very microscopic eruption of the parasites, which have a tendency of coming out. Okay, so that's why there is scratching. Excoriation is basically scratching on the site of extravasation of the parasites of scabies. So thyris pubis, that is pediculus humanus, they will go from outside to inwards, but scabies, my apologies, I'm sorry. Scabies will cause the depositions of the eggs, and then when they move around, they cause the scratch. So excoriation, the word is very particular for scabies. Are we clear about this? Yes or no? Okay. Next one. Next one is 32-year-old male presents to the emergency department complaining of fever, malaise, and a maculopapular rash that includes his palms and soles. When the patient's serum is added to the mixture of cardiolipin, lecithin, extensive flocculation is observed. What is the next step in the management? If anyone is ready, this is a very easy question. They have said everything they have to say over here. Which is, first of all, which test are they trying to describe over here? VDR, very good. Venereal disease research laboratory. What is the answer? C. C is for spirochetal antibodies for syphilis. Okay, last question. A 65-year-old woman with ovarian carcinoma is admitted to the hospital for elective chemotherapy. In the hospital, she has a fever, headache, and diplopia. Broad-spectrum antibiotics are initiated, but the patient becomes progressively more obtunded and is found dead by nursing staff. A week later, an 82-year-old man develops similar symptoms while in the same hospital following knee replacement surgery. The state health department is notified and reports that there have been cases of similar illness. A subsequent investigation reveals a total of 8 affected patients, all of whom consumed dairy products. Wait, you do not even have to read the whole question. The last line, consumations of dairy products, is enough to name the organism from over here. Which organism will spread from dairy products? Okay. Okay, I did not know that you guys were so good at microbiology. How did you guys get so good at microbiology? I mean, it's absolutely breathtaking. Okay, it's absolutely amazing. It's your fault. Okay, no, it's your fault. Okay, it's not my fault. It's your fault that you're so good at this. Okay, it's not my fault. Okay, so good job, guys. Okay. Oh, we have another question. Okay, let's see how good you are with the last one. Okay, no. So good job. The last one was Listeria. Can you guys name another source of contamination with the Listeria organism? Very high yield for Listeria. Deli meat, very good. Not just any meat, meat from delis. Okay, cold meat, very good. Next one, a 34-year-old woman comes to the office due to 3 days of discomfort and swelling in the axilla. The patient has no known medical problems. She is the local community veteran and volunteers as a swim coach at her children's school. She owns a parrot and a hamster. That do you realize? Okay, she owns a parrot and a hamster. Okay, the temperature is 36.9, meaning normal. On physical examination, there's enlarged tender axillary lymph node on the right, measuring about 3, and surrounding erythema. Sounds like a very familiar disease, we know. There is no other area of lymphadenopathy. Several scratch marks are seen. The organism responsible is associated with which of the following carcinoma? Very good question. If anyone is ready, please tell me you're ready. Okay. What is the name of the organism? Bartonella Hensley, very good. What is the name of this disease? Cat scratch disease. And what is the name of the tumor? Bacillary angioimatosis, very good. So her pet cat was harboring Bartonella, and Bartonella Hensley is responsible for causing cat scratch disease that is characterized by scratching lymphangitis. I love how they gave other animals too. Did they? All right. All right, yeah, parrot and hamster. Right, I just wanted to let you guys know how unimportant that one was, because if there was a disease with the parrot, which type of disease or which type of microorganism can the parrot spread? Very good, psittacosis. Which type of disease can the hamster spread? Yes, Yersinia pestis. Okay, so congratulations, guys. 5 out of 5, almost like all times. Okay, right? I'm sure you guys missed doing questions last week because we did not do any questions. So like all times, 5 out of 5, very good. Good job. Really proud of you guys. I really am. With that being said, I would like to end the lecture over here for today. Okay, so you guys should be really proud of yourself. And tomorrow morning, we're going to start the lecture at 9 a.m. Hopefully, if we don't, then I'll send you an email because we're going through a change of duties right now. So now, with that being said, does anyone have any question regarding our lecture? Because one of our team members let us know that some students have questions regarding our lecture. If you do, now is the time for you to ask me. I'll give you the answer right away. Did anyone have any question about our lectures?
Oh, yeah, I just wanted to know when you took your step 1 and what did you score?
When I took my step 1, that was almost 1.5 to 2 years, I guess. And my score was, this is the first time I'm actually revealing my score. I actually never really revealed my score to my students, but it was actually something which I did want. My initial goal was to go into general surgery. So my goal was to score anything above 220. So I was prepared. I got done with my bachelor's and I prepared for 6 months and I got a score of 242. So that's that. Any other question?
Can we repeat the first aid? So for example, if we finish one cycle, can we do it again?
What do you mean by doing it again?
So for example, let's say I pay for one round. Do I have to pay again for the next round?
Okay, that's a good question. So basically, as you can see, that the lecture number for today is the 78th lecture. So that means that it's been 78, that this is the 78th class of the first batch of students. So for the new students who would be starting the lecture again with lecture number 1 from February, let's say you, for example, if you are done with your lectures with us, then whenever you want in the future, if you want to join the lectures again, let's say we will do cardiovascular system with you in our new batch, and then that batch is done. And then we have another batch where we will do cardiovascular system once again. And if we do that, and you want to join the lectures again, you are more than welcome to do so without any transactions. Okay, any other question? Yes, you have a question. What is your question? Dr. Tarian, what is your question? I know you're good at recall. So how did you practice that? Good question. Okay. So what I did was during my dedicated period, so my preparation time was very less. Okay. I prepared for 6 months. I hope I took more time, but I really didn't because to be honest, I'm a bit impatient when it comes to my preparations. I felt like I was okay because to be honest, I did not need a very good money. Like for example, I was not aiming for a score that was exceptionally high, that is somewhere in 260s, 270s, because I know the type of residency I want to go to, and the amount of score I have is more than enough for me to get into that. So I was not willing to waste a lot of my years or time to do that. So what I did was, for my recall, I actually read first aid close to 4 to 5 times, and I was so obsessed with reading first aid that there were times that I remember that I was reading first aid in my sleep. Like I would literally lie down, close my eyes, and just try to remember which page of first aid said what. And if I couldn't remember, I'd wake up and then I try to go and see it. So what happened was, I mean, that's a weird type of obsession which I had. That is not something which I encourage you guys to do the same, but that's what I did. Mine was a lot of repetition. Like I did a lot of repetition. I would study for 8 to 10 hours during my dedicated period, and that's the same way I expect my students to study during their dedicated period. That is, I would wake up in the morning, first thing in the morning, before having a heavy breakfast. I would make sure that I would do 3 sets of questions, of blocks of questions, of 40 questions each. Then I'd take a break. Then I'd read microbiology for 1 hour. Then I would read pharma for 1 hour. And then I would read the rest of the first aid for 1 to 2 hours. So that's almost close to 6, 7 hours of my day gone. The rest of the day, I'll watch 1 to 2 pathoma videos, and then I would solve the questions I missed out. For example, the 3 blocks which I would do in the morning, I would do the wrong questions. So that's what I would do 6 days a week. And I would take 1 day off of my dedicated period to cool down. So that's that. How many NBMEs did you do? I did all the NBMEs. I did not leave out any NBME. I did all the NBMEs starting from which one? Starting from the free ones to the new ones. I did all the NBME. Okay. Was your first, okay, let me see, we have a lot of questions coming. Okay. How many NBME? Was your first aid pass with a tutor or alone? I always studied alone. I studied alone. To be honest, I actually did have a tutor at the beginning, but that tutor was no good because I actually lost a lot of money with that tutor because he was not very helpful. Because to be honest, tutors in this country, that's a weird thing that which I realized is that tutors in this country, they have a habit of charging you 50 to 100 bucks per hour, and they do not read the first aid like how we read the first aid. Do you understand? Like they just tell you what to do, but they don't they don't explain how that should be done. So the way I thought that I would take our lectures is that I would make sure that students do not go through the same issues which I went through. So that's why that. And another one is I personally believe that charging a student, especially a medical student who already has so much burden, more than 100 bucks per hour is absolutely unacceptable. So we tried to keep our pricings as low as $1. I think I already mentioned that in the first lecture. So that's that. So yes, I did it first with the tutor. It did not work. And then I did it all alone. I had another student, friend I was studying with, and we did first aid together once, but that's that. Does anyone have offline pathoma videos? Okay, so that question is not for me. I started with renal with first round of students. When the new session starts, can I continue to use first aid 2020? Yes, of course you can. No problem. Next question. How will we know you are doing that particular topic again, like CVS, for example? When we start the new session, I will tell you which topic I will begin with. We always begin with the organ systems first. So we will begin with cardiovascular systems. So don't worry, I'll send you emails and everything which you need to know. Next question, how many times you finished UWorld? I finished UWorld 2 times, and I finished AMBOSS 1 time. But AMBOSS was not that high yield. UWorld, I wish I did UWorld 2 more times, to be honest. Instead of doing AMBOSS, I wish I did more UWorld because the actual exam was very similar to UWorld. Any other question? Any other question? No. Perfect. Okay. So that's that. Okay. You know, like how you have videos on YouTube saying shortcuts to good score? You know, like shortcuts, 5 things you can do to increase your score and shortcut and this and that. Just to let you know, there are no shortcuts, to be honest, okay? All you have to do is work hard and study first aid, UWorld, and pathoma. No one tells you that. No one really talks about the importance of these 3 things, okay? So keep your calm, don't worry, keep on doing the lectures. And to be honest, I have had students who have, meaning that my students have always scored more than me. I think that's a bigger achievement for me, more than my score, because I personally think my students are always better than. That's why I keep on telling you guys that you guys are so much better than I was, because when I was studying first aid, my answers will not be the same as yours. So that's that. Okay, with that, I wish you guys all the best. Does anyone have any other question? I'm going to ask one last time before I end the lecture. Anything regarding the lecture, anything? Anyone? Any questions? Okay, perfect. So hope you guys have a great day today. If you guys have any more questions, send us an email. I will try to get back to you. And with that being said, hope you guys have a great day. I'll see you guys tomorrow at 9. And thank you so much.